welcome to PW NEET English YouTube channel. I'm Shalini Somashekar, your botany teacher, and in this lecture we are going to be covering the chapter Sexual Reproduction in Flowering Plants in one shot. Now speaking of flowers, can you imagine living in a world without them? Nature would look so boring with just different shades of green, right? It wouldn't look as aesthetic as it does today. Well, we humans use flowers for so many different reasons. We use them in the preparation of floral fragrances, flowers are used as an expression of love, flowers are used as components of home decor as well. While we use flowers in so many different ways, why do you think plants produce them in the first place? That's exactly what we are going to learn in this chapter. This chapter carries high weightage in your NEET exams and you can expect as many as 3 to 4 questions to appear from this chapter. And in order to make your learning journey smoother, I'm going to be sharing the class notes along with some DPP questions that you can solve in the batch called One Shot by Shalini Ma'am. It is a free batch and you can access all of the resources there on the PW app for free, for which the link is given in the description box below. And while you're on the app, make sure you check out the super useful quizzes and study material which many students in the past have benefited from. Now without any further ado, let's get started. Alright, now this is one of the 3 chapters in the first unit of your 12th grade syllabus, which is called reproduction. This is the first chapter. Following this, you have 2 other chapters, human reproduction and reproductive health, both of which will be covered in the zoology part. You know, right, in NCERT, at the beginning of every unit, there is a small introductory paragraph. Since this is the first chapter, before this, there is that introductory paragraph. We'll go through that first and then we'll deep dive into the concepts from this specific chapter. Since this is the first chapter in the entire textbook, in this paragraph, there is a line about biology to start with. We know what biology is. It is a branch of science which deals with life or living organisms. The same thing is mentioned here. It says that biology, in essence, is the story of life on Earth. We know that in biology, we learn about how life originated on Earth, how life evolved, what are the various characteristic features of living organisms, differences, and all of that we learn, right? We know about that. What do we also know? We also know that no living organisms are immortal. All of us at some point will have to die, regardless of which species. The lifespan is limited. It's not forever, right? Some living organisms may have a shorter lifespan spanning a few minutes, and for some of them it could be hundreds of years. Regardless, death is inevitable. All living organisms at some point have to die, right? We know that. If all living organisms are going to die at some point, how is it that some species continue to exist for several thousands and millions of years? The reason why any species continue to exist even though living organisms are frequently dying is because of a process called reproduction. Now this reproduction can be of many different types. What does reproduction mean? Production of something again, right? So reproduction is a process by which living organisms make organisms which we call progeny or offspring, which are very similar to the parents, okay? So by that process of reproduction, more and more organisms of that species get added on. So even after few organisms die, there will be other organisms. They will reproduce and continue to add more and more organisms in that population, and a species will continue to exist, right? There are many different ways in which this reproduction can happen. It can happen asexually or sexually. Regardless of whether it is happening asexually or sexually, it will give rise to newer organisms similar to itself. Now that is what this talks about. Basically, it is because of the process of reproduction that an organism continues to exist on this planet, a species continue to exist on this planet for several years. Now, there are instances where some species have gone extinct, they no longer exist on this planet. That is rare case, right? Dinosaurs used to exist and they no longer exist. There are so many subspecies of tigers that no longer exist. There is a reason why those extinctions happen. They happened either because of some natural cause or because of some man-made cause, which is called the anthropogenic cause. So unless such things happen because of reproduction, a species will continue to exist. Okay, so that's pretty much what is given in this paragraph here. While individual organisms die without fail, species continue to live through millions of years unless threatened by natural or anthropogenic extinction. Anthropogenic means man-made reasons. Like some animals could have gone extinct because humans overexploited them, they overhunted them. For that, they may have gone extinct. So, but otherwise, most organisms will continue to exist. So, over here, reproduction becomes a vital process without which species obviously cannot survive for long. Each individual leaves its progeny by either asexual or sexual means. Both will give rise to offspring, both will give rise to progeny, but sexual reproduction is slightly more advantageous because through sexual reproduction, newer variations are introduced into the population because during sexual reproduction, meiotic cell division takes place. All about that we will learn as we move on. Okay, so this unit is all about that. In this unit, we will learn about how sexual reproduction happens in flowering plants in this very chapter. Following that, you will also learn about how reproduction happens in humans. We will also learn a little bit about reproductive health in humans in two other chapters. Okay, so that is the unit introduction. Now, at the beginning of this chapter, there is some information given about Panchanan Maheshwari, who is one of the most renowned botanists from our country. He was born in the year 1904 in Jaipur, Rajasthan. And the reason he is so widely known is because of his contribution to the field of plant biotechnology in India. So what he basically did was he studied how embryos develop in seeds. Be it monocots or dicots, within the seeds, there is a specific pattern of development of embryo. When fertilization happens, the zygote will form. The zygote will undergo a series of cell divisions and it will give rise to the embryo, which will further develop. The embryo present within the seed is what will become the future plant. Okay, so there are a specific series of events that take place when embryo develops. He studied about that in both monocots and dicot plants across many species, and he noticed how these developmental characteristics did not vary very much between species to species. Okay, he saw that was kind of constant the way the embryo developed. So he thought that this was a reliable factor or a characteristic that can be considered during classification of plants. Taxonomists classify plants into different groups, right? They classify plants and other species into different groups based on some similarities and differences. So while they're trying to classify different organisms into groups, they take into consideration morphological features, certain anatomical features, cytological features. He also insisted that they should consider embryological development also as an important feature to classify plants because that does not change very frequently. Okay, that is one thing. And in the University of Delhi, he founded and established the Department of Botany. In this Department of Botany, a lot of research went on in the field of plant embryology. Okay, so he started to work towards artificial culture of immature embryos. Sometimes embryos do not mature. Embryos are very important in a seed because when you sow the seed, when the seed germinates, a tiny plant comes out of it, right? That plant comes out of the embryo itself. So embryo is like the baby plant that is sitting inside the seed. Sometimes if the embryo is not mature, the seed will not germinate and you will not get the baby plant. Okay, and this may happen even when you are performing artificial hybridization between two different species. Then the seed may not be viable and it may not germinate and give rise to a new plant. That can happen for many reasons. If at all any embryo in a seed is not considered viable, that embryo can be rescued by many tissue culture techniques. So he basically was working towards that. He worked on how immature embryos can be artificially cultured in a nutrient medium in laboratory conditions instead of its natural place, which is inside the seed. Okay, and then he also worked on test tube fertilization. You've learnt about IVF in animals, in humans, right? How male gamete and female gamete are fused outside the body of living organisms in laboratory conditions. He tried to pull off something similar with respect to plants as well, which kind of showed how fertilization does not require intact organisms. It can happen in non-living test tubes as well. Okay, also another important thing was he accomplished intra-ovarian pollination, which got him worldwide acclaim. When pollination happens, the pollen grain specifically goes and lands on the stigma of the flower. The stigma is the place where the pollen can be either accepted or rejected, and eventually, if it is accepted, pollen tube will be allowed to form. Sometimes due to self-incompatibility, or sometimes because the pollen grain belongs to a slightly different species, pollination may not happen. So there are various barriers which can prevent pollination from happening and eventually fertilization from happening. He developed a technique where pollen grains are actually directly introduced into the ovary. Intra-ovarian means within the ovary, pollen grains are being introduced. So in that way, if there is any problem with stigma pollen interaction, pollen pistil interaction, or if the pollen tube is not capable of forming a pollen tube through the style, whatever those problems were, they can be overcome if the pollen grain is directly deposited near or inside the ovary of the flower. Okay, so he performed that. And because of his immense contribution in the field of plant reproductive biotechnology, he won many fellowships, one of which is the Fellowship of Royal Society of London. Also, he was honored with a fellowship in the Indian National Science Academy and many, many more institutions. He is one of the pioneers in setting and putting together the NCERT textbook for the textbooks that we all use right now. Its ancestors, like very, very much before the initial days, at around the year 1964, he was one of the people who brought about the first textbooks of biology for NCERT. Okay, so that is some of his contributions that you will have to remember. Now that we're done with the unit introduction and spoken about the contributions of Dr. Panchanan Maheshwari in the field of botany, we can move on and step directly into sexual reproduction of flowering plants. So now, without any further delay, let's get started. So what do we start with? The name of the chapter is Sexual Reproduction in Flowering Plants. We're not talking about sexual reproduction in plants in general, right? We're specifically saying flowering plants, which indicates that all plants don't produce flowers. You would have learnt about it in 11th grade chapter Plant Kingdom, that in Plant Kingdom you have 5 different divisions. So what are they? You have algae, you have bryophytes, you have pteridophytes, you have gymnosperms, and you have angiosperms. The first 4 divisions, algae, bryophytes, gymnosperms, and pteridophytes, all of them you've learnt in detail exclusively how their life cycle is, how reproduction happens, and all of that in Plant Kingdom itself. But angiosperms, about that you just briefly touched upon. So in this chapter, you will learn in absolute detail how sexual reproduction happens in angiosperms. Flowering plants is another name for angiosperms. You may have noticed how not all plants produce flowers. You don't see algae producing flowers. You don't see mosses producing flowers. Ferns or a Christmas tree does not produce flowers. So flowers are not a characteristic feature of all plants. Flowers are produced only by a specific group of plants that is called angiosperms. Now, these angiosperms are the most advanced and the most dominant kind of plants that you can find on our planet currently. Now, let's get to the reason why these plants produce flowers. The reason they produce flowers is because in them, flowers are the sexual reproductive organs. You know, in humans, testes are called male reproductive organs and ovaries are called the female reproductive organ, because when we talk about sexual reproduction, one hallmark feature of sexual reproduction is the fusion of gametes. Male gamete fuses with the female gamete to form the zygote. The zygote will eventually develop and give rise to the next generation, right? That's what happens. So whichever organ is involved in the production of gametes, that is called the sex organ, right? So likewise, in plants, it is within the flower that the reproductive structures and the gametes and all of that will form. Basically, the male spores and female spores form within the flower. That is why in angiosperms, flowers are the sexual reproductive organs. Okay. Now, you know how in humans, the moment a baby is born, it is not capable of reproduction, right? Same thing will apply even to plants. The moment a plant is born, when can you say a plant is born? When does the life of a plant begin? If you think about it, when the seed germinates, a tiny plant you can see outside, that is when you can say the life has begun for the plant, right? Of course, even inside the seed, the embryo was there. But once the embryo comes out, germinates out of the seed and forms a plant, you can say that a new plant has been born technically, right? So, the moment the seed germinates, have you ever noticed the plant will start producing flowers? It doesn't. There is a period of time where vegetative growth will happen. During that time, the stem elongates, leaves will form, leaves will start photosynthesizing, excess food will get stored in the form of starch. Eventually, only a couple of days or months later will the plant start producing flowers. And the reason is simply because It is through the flower that that particular plant can get its next generation. If you think about it, all living organisms have one biological purpose to serve on this planet. Even after we are gone, even after we die, our species has to continue. So we do everything that is possible by us to make sure that our babies are kept safe, they reach adulthood, they reproduce, and the species continues. We think that we are the clever species, most advanced, most brainy animals to live on this planet. But plants, despite lacking a brain, are so clever. They also know equally well that if they produce children when the conditions around are not favorable, the species may not continue to exist. What are babies for a plant? The babies for a plant is seeds. How do seeds form? Seeds form after flower is formed, flower transforms into the fruit, within the fruit seed will form. So the plant has to be ensured of the fact that in the upcoming time, if the flower is produced right now, by the time the seeds form, will the conditions around it be favorable or not? Plants have many ways of detecting it. They do not have sense organs like us, but they do have certain pigments and hormones that can sense its surroundings. So whenever a plant senses the temperature, the day length and all of that, and makes sure that if flowering happens right now, by the time the seed forms, the environmental conditions will be favorable for the germination of the seed. It will favor the growth of that particular plant. Only then will it produce flowers. Until then, it will wait. Okay. So even before you actually see the formation of a flower on a plant, the plant would have made the decision that it has to flower. How does it do so? It waits for signals from the leaves and other parts of the plant. How are these signals sent? They are sent by means of hormones. There is a particular hormone called a florigen which is produced by the leaves and it gets transported to the apex of the shoot. Normally what happens at the shoot apex? Leaf primordia will form and then at the shoot apex newer leaves will start arising, right? Once the plant has decided that the conditions are right now favorable, the temperature is perfect, the environmental conditions are great, the sunlight is just the right amount, there is sufficient water in the soil. Once they sense all of that and they're like, okay, now is the time to make babies, even if the baby is produced right now, I mean the seed, it will survive very well because the conditions are favorable for its growth. It cares so much about its next generation, right? So once that is confirmed, the chemical signal in the form of this hormone gets sent into the shoot apex. Normally, the shoot apex was producing leaf primordia and eventually that would form leaves. Now that it has received the signal from the leaves, it has to stop producing leaves and start to make floral parts. So, the tip of the shoot will now not produce leaves, it will produce floral primordia. It will transform into a floral meristem. The pattern of cell division at the shoot apex will change a little bit and instead of producing leaves, it will start producing flowers. Okay, so basically, even before the actual flower is seen on a plant, the decision that the plant is going to flower would have taken place much before we see it. When can we see it? The more earliest that you can see it is when the floral primordia forms. Okay, so several hormonal and structural changes are initiated. That leads to the differentiation and further development of the floral primordium. This primordium means like a precursor. Floral primordium is going to give rise to the flowers. Okay. So inflorescences are formed which bear the floral buds and then eventually the buds will transform into flowers. So what is a flower? Flower is a modified condensed shoot. What does this mean? What does modified mean? Modified means, you know, in morphology of flowering plants, you've learnt about certain modifications of leaves, stem, roots and all of that, right? What is a modification? Modification is when a particular plant organ is doing something other than its primary job. If a leaf is doing something other than performing photosynthesis, like if it has transformed into spine, then that is a leaf modification. If a stem is doing something unusual like photosynthesis other than its primary job, then it is called a stem modification. So that way, we say that the flower is a modified shoot. That means shoot normally would produce leaves. Now instead of leaves, it is modified to do something else. It is producing a flower. That is why we call it modified shoot. What does condensed mean? Condensed means kind of compressed. Now see, if it was like a normal shoot, it would have the stem and at the nodes, the leaves would form like this. Now we are saying that this is modified. This is the normal shoot, vegetative shoot. When it modifies to produce a flower, what basically happens is, at each node, instead of leaves forming, modified leaves will form. Instead of these regular leaves, at the lower node, calyx will form. Instead of these leaves, those leaves will change color. Okay, they will have different kinds of pigments, and instead of regular leaves, petals will form. Instead of regular leaves in the further higher up node, stamens will form. Much higher above that, instead of leaves, the pistil will form. So basically, the leaves, the shoot is modified here. It is doing something that it usually doesn't do. An unusual function is being performed here. Why is it being performed? Because to bring about sexual reproduction. So we've understood what modified means. What does condensed mean? Condensed means it's compressed. So basically what happens here is, you know that in the shoot, these points at which the leaves arise are called the nodes, and the region between the nodes are called internodes. So the internodal length here will get reduced and the shoot will appear such that all of these floral parts are arising from a common point. So it's almost as if the internodes are highly reduced in length. So eventually when you look at it, it will appear like this. Now it will seem like everything is arising from the same node, but that is not the case. What has actually happened here is they are arising at different nodes, but the internodal length is so condensed, so shortened that it will appear like all of them are arising from the same point. So this is how a flower forms from the shoot. Shoot gets modified and condensed, internodes will condense, and that is how the flower is formed. Now, we are talking about sexual reproduction in flowering plants. I've already mentioned earlier the hallmark characteristic feature of sexual reproduction in higher organisms, higher plants and animals is fertilization. Male gamete fuses with the female gamete to give rise to the zygote. The zygote will eventually develop to give rise to the next generation. Fertilization happens even in angiosperms. In fact, the fertilization in angiosperms is kind of unique compared to the other groups of plants. More about that we'll discuss as we move on further in this chapter. So here we will talk about fertilization. Basically, this chapter is split into three different parts or three different blocks. We will learn about fertilization as well. That is one part of this chapter. The second part of this chapter, before fertilization, whatever structures are formed in a plant, whatever events take place in a plant, that we will learn under pre-fertilization structures and events. Pre means before. So what all happens inside a plant before fertilization, all of that we learn about here. Next, in the third part, we will learn about post-fertilization event. Post means after. Following fertilization, after zygote is formed, what will the zygote develop into? What will happen to the flower? How is the fruit formed? All of that we will learn under post-fertilization structures and events. Okay, now let's get started and talk about the structure of a flower. We've learned about it in the chapter Morphology of Flowering Plants in 11th grade, but let's get like a quick recap on the different parts of a flower. Flowers come in so many varieties. Some of them don't have some parts that I'm going to draw right now. Some of them may have additional parts that I will not mention right now, but what I'm going to mention right now is only like the typical flower which most of the angiosperms produce, okay? Of course, there will be slight modifications from what I'm going to draw here, slight variations you can see, but this is just like a generalized flower diagram, okay? So some flowers have a stalk, some don't have a stalk, but most of them have a stalk. So the stalk of a flower is called the pedicel. At the tip of the stalk, you will have a broad region which is called thalamus. On the thalamus, the four different plant parts that I showed you right here will be arranged in the form of concentric circles. Okay, so you can see if this is the thalamus, if you look at it from above, if this is the thalamus, on top of the thalamus, the outermost whorl or outermost circle will be made up of calyx or the sepals. Inside that, you will have the whorl made up of petals. Just give me a moment. It's arranged in the form of concentric circles like this. Okay. Okay. So this here is the thalamus. On top of the thalamus, floral parts will be arranged in the form of concentric circles, and those concentric circles are called whorls, WHORLS. Outermost whorl, you will have sepals. Okay, so you will have sepals present here. These sepals together constitute what is called calyx. The function of the sepals is to protect the flower when it is in the bud condition. What is present in the second concentric circle from the outside are petals. Okay, so inside that you will have petals present, and collectively all the petals together is referred to as corolla. Inside the petals you have the male reproductive part, which is referred to as the stamen. That is the third whorl from outside. These stamens are collectively called Androecium. And right within innermost whorl is your pistil or gynoecium. Okay, so its individual units are called carpels. Collectively, it is called the pistil or gynoecium. Its units are called carpels. Together, you call it either the pistil or gynoecium. Over here, stamens are the male reproductive part. Pistil is the female reproductive part. Okay. Both of them are absolutely essential for the process of sexual reproduction because it is within the stamens that the male spores get formed. It is within the pistil that the female spores will form. Without them, sexual reproduction cannot take place. But without the sepals or petals, sexual reproduction can still take place. They are not absolutely essential for sexual reproduction, but if they are there, obviously it is beneficial. The sepals will protect the inner parts present inside the flower in the bud condition before the flower has bloomed. And the petals, which are generally brightly colored, if they are brightly colored, they are colored because to attract the pollinators towards them for pollination. But even if that doesn't exist, those whorls don't exist, reproduction can still take place. Now, because stamens and carpels are absolutely essential for reproduction to take place, they are called essential whorls. The other two whorls that may or may not be present, even if they are not present, if the function will still happen, such whorls are called non-accessory or, accessory or non-essential whorls. You've learnt a lot about petals, sepals and its variations in the chapter Morphology of Flowering Plants. So for now, since this chapter specifically is about sexual reproduction, we will focus mostly on stamens and carpels, its structure and all of that. Okay, now let's get started with the stamen. An individual unit of Androecium is called the stamen. The stamen, as you can see here, is attached to the thalamus on one end. So, a stamen has two important parts. It has this long, slender stalk, which is called the filament. At the tip of the filament, you have a knob-like structure, which is called the anther. The part of the filament which is attached to the thalamus, this end of the filament is called the proximal end. It is close to the thalamus. Proximity is closeness. This is proximal end of the filament. On the distal end, it bears a knob-like structure which is called the anther. again in the number of stamens, whether or not they are free, whether they are adhered to the stamen or adhered to the petals or the tepals. You have many different types. Its length varies from species to species. More about that you've learned in morphology chapter. For now, let's consider a typical angiosperm anther. In a typical angiosperm anther, you will find two lobes. Okay, there are species where a single lobe is also present, but in most cases there are two lobes present. You can see there's one lobe here. You can see over here, this is one lobe, and here you have the other lobe. There are two lobes present. Because an anther has two lobes, because anther has two lobes, it is said to be bilobed. Bi means two. Now, when you take an extremely thin section of the anther transversely, if you cut it like this, take a section and look at it under the microscope, or just cut it in half and look at it from above, you will be able to see there are two lobes here. This is one lobe and this is the other lobe. If you take a look at each lobe, now let's look at just this one lobe. This one lobe. You can see that this lobe has two chambers present inside. This is one and this is the other. There are two chambers or cavity of sorts which are present inside each lobe. These are called theca. It is within these theca that pollen grains will form. This theca runs throughout the length of the anther. It's like inside your pen how there is a refill. It's a tubular structure, inside that there's another tube. Similarly, you have the the anther wall. Inside the anther wall, there is a region where pollen grains will form. So that space or that chamber is what we call theca. Now that each lobe has two theca, the anther is said to be dithecous. Di means two, bi also means two. These are important terms that you'll have to keep in mind, dithecous and bilobed. Now, these are regions where microspores or pollen grains will form. Remember, in plant kingdom, you've learnt about what are structures that produce pollen called. A structure that produces or gives rise to pollen grains, the structure that gives rise to microspores are called microsporangia. Remember, we've discussed sporangia in the past as well. Any structure that gives rise to spores is called a sporangium. Over here, within these theca, microspores will form. If microspores are going to form within this, what does the structure become? The structure is called a microsporangia. If you look at it, each anther has 1, 2, 3, 4 microsporangia. Since each anther has 4 microsporangia, it is said to be tetrasporangiate. Okay, tetra means 4 here. It is said to be tetrasporangiate. Remember, dithecous is for each lobe. Okay, one anther, two lobes, bilobed. Each lobe has two theca, dithecous. Again, in an entire anther, you have 4 sporangia, so tetrasporangiate. These are terms that you need to remember and understand, because based on this, application-based questions can be asked in your NEET exams. Okay, now let's move on. Let's talk about the structure of one microsporangium here. If you see, this is a transverse section of a young anther. You can notice here clearly how there are two lobes. This is one lobe, this is the other lobe. The two lobes are held together by a tissue called the connective. Right here at the center is the region where microspores will form. Okay. So there are 4 microsporangia here. Okay. So this is one microsporangium. This is one microsporangium. This is one microsporangium. This is one microsporangium. So in each microsporangium, there is a central tissue which will eventually give rise to the microspores. Microspores are very, very important cells for the plant. It is very precious cell for the plant because microspore itself will eventually produce the gametophyte, and the gametophyte has to produce gametes. Without gametes, sexual reproduction cannot take place. So the region where microspores are getting produced, there will be so much protection. Okay. So in these microsporangia, there are thick walls present. This is the wall of the microsporangia. Inside the wall of the microsporangia, thick wall, there are tissues which will eventually give rise to the microspores or pollen grains. So let's just consider one microsporangium and see what are the various layers present in its wall. So here you can see this is the outermost layer in a microsporangium. Basically, the wall of the microsporangium has 4 different layers. Like all parts of the plant, leaf, root and stem, the outermost layer is called the epidermis. Even over here, the epidermis is a single layer of cells. You can see the epidermis here. Okay. So this is the epidermal cell layer. Outermost layer here is the epidermis. Obviously, it's going to offer protection to the developing microspores. Okay. So epidermis is protective in function. This epidermis is made up of a single layer of cells. It is protective in function. Now, following the epidermis, in the immediate layer below the epidermis, you have what is called the endothecium. Okay. So, right beneath the epidermis, you have the endothecium. This endothecium also is one layer in thickness. Okay. Now, following the endothecium, you have the middle layers. Middle layers can be around 1 to 3 cell layers in thickness. Okay. So, this is 1 to 3 layers in thickness. Following that, the innermost layer, maybe which color do you think I should take? Maybe pink. The innermost layer here, this layer is called tapetum. The tapetum is also generally single layered. Right inside the tapetum, whatever you're looking at here, right inside, this is the actual cells that are going to give rise to the microspores in future. These cells are called microspore mother cells. The entire tissue is called sporogenous tissue. Sporogenous means what? Spore generating. Sporo for spore, microspore, genus generating. So this is sporogenous tissue. Now, we know about the structure. Outermost epidermis, inside that you have endothecium, inside that you have middle layers, inside that you have tapetum. Now, it's important for us to understand the functions of these different layers in the wall. Similar to how our walls in our house, they are protective in nature. Whatever is present inside, we all live inside. It protects us from the harsh climate outside, from the animals living outside. It offers protection. Same way, the wall of the microsporangium also offers protection to whatever is lying inside, the precious cells which will eventually give rise to the microspores, right? But different layers perform different functions. Even in our houses, we have walls of different layers. There's a brick wall inside which provides the framework. Outside that, there could be like a cement layer. On top of that, there could be a primer. On top of that, there could be paint. On top of that, there could be a wallpaper. So you have many layers. Each layer performs its own function. So overall, their function is to offer protection, but every individual layer does its own thing there. So the outermost 3 layers, these 3 layers, epidermis, endothecium and middle layers, these perform different jobs at different times. If the anther is young, when the anther is young, it offers protection. Right now, it's going to protect the cells inside, which is eventually going to give rise to the microspores. But once microspores are formed, pollen grains are formed inside the anther. Pollen grains have to be released. If these three layers act like a rigid layer providing too much support to the developed pollen grains, it's offering too much protection, it's being overprotective, then the pollen grains cannot get released outside and bring about pollination, right? So when the pollen grains are mature, the wall layers realize that it's time to let go. The same three wall layers that were offering protection up until this point will start helping the pollen grains get released outside by breaking themselves apart. Okay? So the process by which pollen grains are released outside the anther is called dehiscence. So once the pollen grains are formed inside, once the anther matures, the layers help in dehiscence of the anther. Okay, similar to how your parents behave with you. Once you're like really small, you don't know what decisions to make, you don't know life very well, they're like super protective about you. Once they realize that you're mature enough to make your own decisions, they will give you that freedom, right? You can think about it similarly. So the same three layers that were offering so much protection, once they realize that the anther is mature, the same layers will break apart and help in releasing the pollen grains from the anther. The process is called dehiscence. So when the anther matures, they help in dehiscence. Dehiscence means release of pollen grains. Talking about tapetum. What is the function of tapetum? See that tapetum is lying almost very close to the sporogenous tissue or the microspore mother cells. When a cell has to undergo cell division, a lot of energy will be consumed in that process. So what these tapetal cells do is, they will secrete certain nutritive substances into the sporogenous tissue that these cells can make use of as nutrients. Okay, so these tapetal cells, unlike the other cells in the anther, they can sometimes be multinucleate. They will have a dense cytoplasm, they will have more than one nucleus. So they are multinucleate. They help in nourishing the developing pollen grains or the developing microspores. Now you have the microspore mother cells present inside. The name itself indicates what its job is. It is mother of microspore. That's why it's called microspore mother cell. What does a microspore mother cell do? It will undergo division to give rise to microspores, right? So these are cells that will produce microspores. Microspores are your male spores, right? Now, let's understand how exactly the process of formation of microspores happens inside the anther. I showed you the structure of microsporangium. Within the microsporangium, microspores have to form. The microspore mother cells or the sporogenous tissue cells are directly involved in the formation of microspores. So over here, I will show you just those cells. If you think of this as the sporogenous tissue, let's say this is the sporogenous tissue. You have many cells present within the sporogenous tissue, right? You will have many cells there. Okay. We will see what happens to one cell here. Whatever happens to one cell, the same thing will happen to all of the other cells as well. So we will take just one cell out of here. This cell is called microspore mother cell, in short MMC, or they may be even called pollen mother cell, which is PMC in short. Now these cells are diploid in nature, which means they have two sets of chromosomes. What happens to these microspore mother cells is, normally since they are neighboring cells, in between the neighboring cells in plants, there will be cytoplasmic connections called plasmodesmata existing. Before microsporogenesis, the process begins, they will form very thick walls and get rounded off inside by cutting off plasmodesmatal connections. Whatever connections they had between neighboring cells, it will get cut off because each cell will produce a very thick wall inside made up of a substance called callose. All of the plasmodesmatal connections will get cut off. Okay. Eventually, after that, these microspore mother cells will undergo meiotic cell division. You know, meiosis is reductional cell division. If a diploid cell undergoes meiosis, it will give rise to haploid cells. When one cell undergoes meiosis, it will give rise to 4 cells. So, from this one microspore mother cell, we will obtain 4 cells. Okay. Just draw a thick wall around this because that shows that it is callose thickening. Okay. Now, inside this callose wall, from this one cell, 4 cells will form. Okay, you can see 3 cells here, there's one here, there's one here, there's one here, there's one at the back. Okay. So, totally 4 haploid cells will form. Now that there are 4 haploid microspores formed, they are together. This is called a microspore tetrad. Tetra means 4. Okay, so this is called a microspore tetrad. Within a microspore tetrad, you will have 4 microspores that are haploid. So there are 4 haploid cells present within this microspore tetrad. Eventually, the same tapetum that was nourishing these developing microspores so far will produce an enzyme called callase, and that will dissolve the outer wall of callose present here. Once callase is secreted, that outer wall will get dissolved. Callase will break down callose, and then the 4 cells that were formed will separate. These are now referred to as microspores or pollen grains. These are also haploid cells. Obviously, if these are haploid cells and they're just separating, nothing else is happening from here to here. They're not dividing again or anything. They were just together within the same callose wall. Now that the callose wall is broken apart, they have gotten separated. So they are also going to be haploid. You know, right? In plants, spores are haploid. So these spores are haploid. Now, if you remember the life cycle and alternation of generation in plants, you know that spores will give rise to the gametophytes, right? So these microspores are basically male spores, and this microspore, male spore, eventually has to give rise to the male gametophyte. So that is what will happen in future. Okay. Now, this is like a more digital diagrammatic representation of what I just showed you. Okay. Yeah. Let's quickly go through what we've learned about alternation of generation in plant kingdom. In angiosperms, the major plant body is the sporophyte. All of the cells of the sporophyte will be diploid. The root, stem, leaf, flower, epidermis, all of that is diploid. Within the sporophyte, sporangia will be present. Sporangia will produce spores. We spoke about microsporangia right now. So the microsporangia are present in the anther. Within the microsporangia, microspores will form. So, during the formation of microspore from the sporophyte, meiotic cell division will take place. Now, this spore has to develop into the gametophyte. Microspore is a male spore that will develop into a male gametophyte. The male gametophyte here is nothing but pollen grain. Most people think that pollen grain is the male gamete. Pollen grain is not the male gamete, it is the male gametophyte. So what happens here? How exactly does this spore that is formed here transform or develop into the pollen grain? We'll understand that now. Before that, what are pollen grains? Pollen grains are nothing but the gametophytes of males. I've already told you that they vary a lot in shapes, but most of them, majority of them are spherical in shape. They vary in size as well, but the range lies between 25 to 50 microns. So that's just the general characters of pollen grains. Now, let's understand the structure of pollen grain. When the microspore formed, it had one haploid nucleus, right? This is the microspore. It had one haploid nucleus. As it develops into the pollen grain, basically what happens is, it will produce additional wall layers. So the pollen grain, as opposed to the microspore, will have two layers in its wall. So the inner layer is called the intine, and it will have an outer layer which is called the exine. Okay, inner intine, outer exine. They both turned out to be the same size, my bad. They're right next to each other. Okay, there's not too much of a gap between them, but for your understanding and visibility, I'm showing it like that. The inner wall is known as the intine. The intine is made up of cellulose and pectin, like most plant cell walls. Intine is a continuous layer. Okay. The outer layer is called exine. Exine is outer layer. Intine is the inner layer. Exine is made up of a substance called sporopollenin. Now, this sporopollenin is a highly resistant organic material. It is one of the most resistant biological materials that you can find. This sporopollenin will not degrade even by any enzymes. Even when you treat it with very strong acids or alkali, even at high temperatures, it will not denature at all. It is because of its highly resistant nature that pollen grains are preserved very well as fossils. Okay, so that's one important thing that you should know about sporopollenin here. Now, this sporopollenin in the exine does not form a continuous layer. Unlike intine, it is discontinuous. What do I mean when I say it is discontinuous? I mean to say there are certain regions in the exine where sporopollenin is not deposited. There are regions where sporopollenin is absent. And such regions are called germ pores. The regions in the exine where sporopollenin is not there, that is called germ pores. Again, if this is the male gametophyte, inside this the male gamete is going to form. Male gamete is a super precious cell, very valuable, priceless cell for plants, because it is through that sexual reproduction will happen and the next generation will arise, right? So that needs to have a lot of protection. That is why the exine is a highly resistant material. But again, if you're completely covering it and overprotecting it, it may not be able to release the male gametes and make it reach the female gamete. So some amount of freedom has to be there. It is protective, but it gives it some amount of freedom as well through these germ pores. through the germ pores, the male gamete that is produced inside can reach the female gamete. Okay. So these exine can sometimes even have certain ornamentations also present, like in the form of spines, in the form of grooves, and different kinds of ornamentation you can find in some species of pollen grains. Okay, so you may see this in exine. Now, as the microspore develops into the pollen grain, first mitosis division will take place. Remember, it is already a haploid cell. In haploid cells, further meiosis cannot take place. When mitosis takes place in the microspore, first the first mitosis will be unequal, which means it will produce cells of two different sizes. The two cells that will get produced after mitosis will not be of equal size. One will be super large, one will be very small. Okay. So the large cell that is produced here is called the vegetative cell. Let me use a different color. Okay, so this is called the vegetative cell. The vegetative cell is larger. It will give rise, it will have a dense cytoplasm and a very prominent nucleus. The small cell that is produced here, this is called generative cell. When a pollen grain contains these two cells, one vegetative cell and one generative cell, the pollen grain is said to be of two-celled stage. I mentioned that the pollen grain is the male gametophyte. It is supposed to produce gametes. Now, if you look at this, which of these two cells is the gamete? None of them, neither of them are gametes. The gamete has still not formed. So, in a two-celled pollen grain, you don't yet have gametes formed. Eventually, what happens is, this generative cell will undergo further cell division, another mitosis. This will undergo another round of mitosis, okay, to produce two cells. This generative cell will divide to give rise to two male gametes. How many male gametes? There's two of them. They're both haploid. How did they form? They formed when the generative cell underwent mitosis. When the generative cell undergoes mitosis and gives rise to two male gametes, you then have a total of three cells, one vegetative cell and two male gametes. This stage of pollen grain is called the three-celled stage. Now, this vegetative cell is present both in the two-celled stage as well as the three-celled stage. It has a prominent nucleus, dense cytoplasm, has a lot of reserve food. Basically, it is nutritive in function. Within the vegetative cell, the smaller spindle-shaped generative cell will float in the two-celled stage. Once the generative cell undergoes mitosis and produces the two male gametes will float in the cytoplasm of the vegetative cell. Okay? Compared to the size of the vegetative cell, the gametes and the generative cell are much, much smaller in size. Now, this is the three-celled stage. This is the final mature pollen grain. A pollen grain is called the gametophyte. Unless it produces the gamete, we can't say that it is a mature pollen grain. So, the pollen grain has a two-celled stage which is not mature yet. The three-celled stage is the complete mature pollen grain. But some plants don't wait until the pollen grain is in its three-celled stage to break open the anther and release them. In majority of the plants, in more than 60% of the plants, pollen grains are released in its two-celled stage. In the remaining roughly 40% plant species, the pollen grains get released in its 3-celled stage. So, if it is getting released at its 2-celled stage, the second mitotic division will happen after pollen release, probably before or immediately after the pollen grain has landed on the stigma of the flower. Okay. Now, let's quickly recap what we've learned. This process is called microsporogenesis. Genesis means generation. Microspore is getting generated or produced in this process. So, we call it microsporogenesis. Every time a spore is getting formed in plants, meiotic cell division will take place. Angiosperms' main plant body is the sporophyte. So, diploid microspore mother cells will undergo meiosis to produce haploid microspores, which are initially together, 4 cells together in a common wall, that is called the microspore tetrad. Once they get separated, you call them microspores. These microspores are haploid. From one microspore mother cell, you get a total of 4 microspores. And these 4 microspores further go on to develop into the gametophyte. These microspores will undergo mitotic division twice. The first mitotic division will be unequal. Spindle will form asymmetrically. As a result, you will get one large cell, which is the vegetative cell, and a smaller spindle-shaped cell called the generative cell. In the second mitotic division, vegetative cell will not undergo mitosis. Only the generative cell will undergo mitosis. G for generative cell, G for gametes. So, the generative cell undergoes the second mitotic division to give rise to the 2 male gametes. And with that, we will get our functional 3-celled pollen grain, which is mature. Okay. Now, more about its structure and how the vegetative cell and generative cell is, I have added in these slides for your reference. Now, this is the NCERT diagram. You can see how the asymmetric spindle has formed here. This is the microspore. It will enlarge as it grows bigger in size. Vacuolation will happen in plant cells. Following that, it will form asymmetrical spindle, which means eventually following this unequal mitotic division will happen. You have a large vegetative cell and a small spindle-shaped generative cell. That's about it. Now, there are some advantages and disadvantages of pollen grains to us, not to plants. For plants, they are extremely beneficial because it is through pollen grains that the next generation will arise, right? So, what are some of the disadvantages we have because of pollen grains? Pollen grains are tiny, tiny microscopic structures. In most cases, it is produced and released in the form of dust. So, many people with, hyperactive immune systems, they tend to get allergies because of pollen grains. Especially there's this one plant called Parthenium, which is also called carrot grass. In Kannada, we call it Congress Geda. So that plant produces pollen grains, and in the season when that plant produces pollen grains, a lot of people get allergies. Okay. So Parthenium or carrot grass causes allergies in a lot of people. My God, why did this become so fat? Let's change its size. Okay. Now, apart from that, other species can cause respiratory disorders, allergies, bronchial afflictions, asthma, bronchitis, all of that can be caused because our body's immune system, sometimes if it is hypersensitive, then it will recognize that as foreign pathogens, as foreign antigens, and then start giving like a crazy immune response. Because of that, some people get allergies. Now, what is the advantage that we have from pollen grains is that it is rich in nutrients. Because it is rich in nutrients, it is sold in the markets in the form of tablets and syrups, and they are consumed as supplements, nutrient supplements. Now, because of its high nutritional quality, apparently sports people, athletes, and even race horses are fed with these pollen tablets in order to enhance their performance. Okay, so it is claimed to increase the performance of athletes and race horses, so they are fed with this. So these are some of the advantages and drawbacks that we as humans have because of pollen grains. Okay, now what is the job of a pollen grain? The job of the pollen grain is that within the pollen grain, the male gametes are present. This pollen grain has to get transferred to the stigma. Inside the female part of the flower, over here seeds will be there. Inside the seed or the ovule, female gamete will be present. The male gamete here should go and get deposited here. It should form a tube, go and release the male gamete inside the ovule. That is the ultimate job of the pollen grain. But pollen grains don't retain this ability to form a pollen tube and bring about fertilization forever. In some species, the pollen grains lose that ability very quickly. In some species, that ability can be retained up to a few months. Now, the period during which the pollen grain has the ability to form a pollen tube and release the gametes and bring about fertilization, that is called pollen viability. Okay, so the viability period depends on a lot of things. It depends on the outside environmental temperature and humidity, and it varies from species to species. In species such as grass and wheat and other grasses, wheat, rice and other grasses belonging to the Poaceae family, within 30 minutes itself, the pollen grains will lose the ability to bring about fertilization, form a pollen tube and bring about fertilization. So there are some families like Rosaceae, Solanaceae, Leguminosae in which the pollen grains even few months after they are released will still retain the ability to germinate and bring about fertilization. Okay. Now, in them, the pollen viability can last up to a few months. If there are plants which produce pollen grains with very short period of viability like rice and wheat, if you want to increase its viability period, then you could store them in extreme low temperatures in pollen banks basically. So what they do there is they preserve or conserve these pollen grains in liquid nitrogen at minus 196 degrees Celsius. This is called cryopreservation. By doing so, they can store the pollen grains and retain their viability period for a longer duration. Why would anybody want to do that? If there's a plant breeder who wants to produce new hybrids, then for the artificial hybridization experiment to take place, some time is required. Until then, if the pollens have to be stored, they may be stored in these pollen banks. Okay, so that's about pollen viability. Now, what all have we covered so far? We spoke about how microspores got produced. We spoke about how the microspore developed into the male gametophyte. We saw how within the male gametophyte, male gametes formed. Right now, the male gamete is ready. We have to learn about what happens with respect to the female part of the plant, which is the pistil. Once the female gamete is also ready, then we can discuss how that one will mate this one and bring about fertilization. Okay? Now, let's talk about the female part of the flower, which is the pistil. The pistil or gynoecium, the units of it is called the carpel. Carpels can be one in number or many in number. It differs from flower to flower, from species to species. So, if there is only one carpel present, such a gynoecium or pistil is said to be monocarpellary. Mono means one. If there is more than one carpel, then the gynoecium is said to be multicarpellary. Multi means what? Multi means many. More than one is many. Right. Once there is more than one, the number of carpels is more than one, the carpels may be fused with each other or they may be present separately. If they are fused, then you call it syncarpus. If they are free, then you call it apocarpus. Okay? Apocarpus is when it is free. Syncarpus is when it is fused. When the carpels are fused. Examples for that, you have Michaelia. Michaelia champaka is the Sampige flower. Champaka, I think it is called in English. So, in this you can see how the carpels are separate. They are not fused together. In Papaver, opium, you can see that the carpels are fused. So, the ovary here is syncarpus. These are examples that you will have to remember. Now, what are the different parts that you can find in the gynoecium or pistil of a plant, of a flower? You can find three parts. The top part of the flower is called the stigma. When the pollen grains are released, it is at the stigma that they have to come and land. Okay. So, this is kind of like the landing platform for pollen grains. Following stigma, you have a long, slender structure that is called the style. The style in some species is hollow like a tube, in some species it is solid. The style basically connects the stigma to the ovaries. Okay. The third part here is the ovary. Ovary is the bottom part, it is basal, it is kind of bulged. Within the ovary, you have space or a chamber that is called like the locule of the ovary. Inside that, you will have a cushiony structure called placenta present. Attached to the placenta, since this is the female part of the flower, inside this megasporangium or the female sporangium will be present. So inside this, you can find many megasporangia present. These megasporangia are also called ovules. Within the ovary, ovules are present. Within the ovules, megaspores have to form by a process called megasporogenesis. So here, this is a clearer image of just the ovary. This is the wall of the ovary on the outside. Inside chamber, inside you call it the locule. At the center, you have a cushiony sort of placenta present. Attached to the placenta, you have very many ovules. These ovules are nothing but megasporangia. It is within these that megaspores will form. You've seen how fruits in some species have a single seed, in some species fruits have many seeds. What happens is when fertilization happens in a flower, this ovary itself will transform into the fruits. The ovules within will transform into the seeds. So if there are more ovules present in the ovary, then such fruits will have more seeds. If there is only one ovule present inside the ovary, then such fruits will have only one seed. Okay. So the number of ovules may vary. It is only one in wheat, paddy and mango. It is many in the case of papaya, watermelon and even in orchids. That's about the different parts of the gynoecium. Now, similar to how we studied the structure of one microsporangium earlier, we will have to learn about the structure of one megasporangium or ovule. I told you how ovules are attached to the placenta present inside the ovary, right? Now, similar to how leaves and flowers have stalks, the ovule also has a stalk of its own. The stalk of the ovule is called the funicle. Okay. So, this is the stalk of the ovule. This is the stalk of the ovule and the stalk is called the funicle. Now, what I'm going to outline right now is the body of the ovule. This is the body of the ovule. Okay, so this outer lining is the body of the ovule. Normally, how would you see a flower? This will be the stalk and this will be the flower. This will be the stalk and this will be the leaf. Over here, the stalk is here. The ovule is not like this, but the stalk kind of fuses with the body and the ovule kind of inverts itself like this. Okay. Such a type of ovule is called anatropous ovule. There are many different types of ovules based on how they are positioned with respect to the stalk. In most angiosperms, you will find an inverted ovule where the ovule is ultafied, inverted. Okay. So, when the ovule is inverted like that, when it is anatropous, you can see how the body of the ovule fuses with the stalk, right? Because it is bent like this. Now, the placenta will be here. This is the point of attachment of the ovule to the placenta. This is the stalk. This is the body. The stalk has fused with the body. The region where the stalk has fused with the body is called the hilum. Just like how in microsporangia there were wall layers that were protecting the cells that are present inside, even here you have protective layers. They are called integuments. There are two integuments. This one that I have highlighted right now, outlined in orange, that is the outer integument. Inside that, you have the inner integument. Both outer and inner integuments are protective in function. Now, if you notice, they do not fully cover whatever is lying inside. There is a small opening where the integuments haven't fused. This small opening, tiny opening, micro opening is called the micropyle. Like I always say, protection is important, but some level of freedom is also important. If this was completely covered and inside the female gamete was produced, then the male gamete would have no way to reach the female gamete. There should be some gap left so that the male gamete can reach the female gamete. Okay. So that is one of the things the micropyle does. It allows space for the pollen tube to enter and release the male gametes. Okay. Now, that is the micropyle. Right within the two integuments, you have a region called the nucellus. What I am outlining right now in orange, that is the nucellus, not nucleus, this is nucellus. Nucellus is a homogeneous mass of parenchyma-like cells. It is within the nucellus that the female gametophyte or the female spore will form. More about it we will learn in the future. For now, just assume that within the nucellus, the embryo sac or the female gametophyte will form. Okay. Now, the part of the ovule where the micropyle is present is called the micropylar pole. The part of the ovule away from the micropyle is called the chalazal pole. The chalazal pole represents the base of the ovule. If it was not inverted, then the chalaza would be here, right at the base. Now that it is inverted, it is appearing here, but chalazal end is considered to be the base of the megasporangium. Now, inside the megasporangium, right here is where the megaspore will form. How exactly the megaspore forms, we will understand now. The process is called megasporogenesis. The generation of megaspore is called megasporogenesis. Now what happens here is, let's say this is the nucellar region. Within the nucellar region, you will have many cells present. Many nucellar cells will be present here. I told you it's a parenchyma-like tissue. So within that you will have many cells. Since this is a part of the sporophytic plant body, you will have many cells that are diploid here. Okay, so you have a lot of cells. Let's say this is the micropylar end and this is the chalazal end. One cell from the micropylar end, usually hypodermal in origin, just below the epidermis, one cell will differentiate. It will start appearing bigger. It will have a dense cytoplasm and prominent nucleus. That cell will transform into the megaspore mother cell. Previously, we saw how the microspore mother cells were many in number. All of the cells, every one of the cells present in the sporogenous tissue transformed into the microspore mother cell, underwent meiosis to produce microspores. Over here, only one of these many cells will transform into the megaspore mother cell. Only that one cell will get differentiated. Usually it is towards the micropylar end. Now what happens to this megaspore mother cell is, it will undergo meiotic cell division. After meiosis, from one megaspore mother cell, you will get 4 megaspores that are haploid. These are diploid cells. You get 4 megaspores that are haploid. Usually, this tetrad is arranged linearly. Okay. Now, among these 4 megaspores, all of them will not remain functional. 3 of them, usually towards the micropylar end, the ones towards the micropylar end, will degenerate. And only this one remains functional. This one functional megaspore will further give rise to the female gametophyte. The ones that degenerate are generally towards the micropylar pole. The one that remains functional is in most cases towards the chilazal pole. Again, there are variations in this. It will vary from species to species, but this is what we see majorly. This process by which a megaspore mother cell undergoes meiotic cell division to give rise to 4 haploid megaspores is what we call megasporogenesis. Now what happens to the megaspore? Let's understand. Let's go back to the slide and see what's happening here. Within the ovule, the megaspore was formed by a process called megasporogenesis. This is the female spore. This female spore will eventually have to give rise to the female gametophyte, and within the female gametophyte, the female gamete has to form. That is the story, right? So we just saw how one megaspore mother cell gave rise to 4 megaspores, of which 3 degenerated and 1 remained functional. What is the story of that one functional megaspore? We'll understand now. Okay, just give me 1 minute, not 1 minute, 1 second. Some slides are repeated. Let's move them. So we start off with one functional megaspore. This megaspore is haploid. This will undergo 3 rounds of mitotic division. Okay. So first mitosis will happen. When mitosis happens, you know that first phase is karyokinesis. Following karyokinesis, cytokinesis should take place. But over here, after karyokinesis, cytokinesis will immediately not take place. Karyokinesis will happen, nuclear division will happen. It's not immediately followed by cytokinesis. So once this megaspore undergoes the first mitotic division, the 2 nuclei will form and they will form a 2 nucleate condition. Or a 2 nucleate stage, let me say. The 2 nuclei that are formed here after karyokinesis will move towards the opposite poles. Immediately after that, they will not undergo cytokinesis. Again, another round of mitosis will take place. Even here, when mitosis takes place, karyokinesis will occur. Immediately following that, cytokinesis will not take place. As a result, in this gametophyte, you can find the formation of 2 nuclei at each poles. Right now, there are 4 nuclei. We call this the 4 nucleate stage. Following this, another round of mitosis will take place. And when the third round of mitosis takes place again there will be karyokinesis, but no immediate cytokinesis. Therefore, the embryo sac will have 4 nuclei at each pole. So, totally there are 8 nuclei. This is called 8 nucleate stage. Following that, karyokinesis, will no longer happen. At this point, cytokinesis will happen and also reorganization will happen such that One nucleus from each pole will move to the center, and around each nucleus, a cell wall will form. Okay. So now when this happens, you can see if one nucleus from each pole move to the center, you will have 3 nuclei at this pole, 3 nuclei at this pole, 2 nuclei at the center. Now that cytokinesis is happening, at this pole you will have 3 cells, at the opposite pole you will have 3 cells, and in between these 2 nuclei, a cell wall will not form, both of them will remain in the same cell. So at this point, you will have a total of 1, 2, 3, 4, 5, 6, 7 cells and 1, 2, 3, 4, 5, 6, 7, 8 nuclei. This embryo sac is said to be 7-celled and 8 nucleate embryo sac. This is the mature embryo sac. Female gametophyte is also called embryo sac. I hope you have understood. After megasporogenesis, the functional megaspore towards the chalazal end in most cases will undergo 3 cycles of mitosis. Initially, karyokinesis will happen, but cytokinesis will not immediately happen. Cell wall formation does not happen. So it remains multinucleate until 8 nuclei are formed. After that, reorganization of the nuclei and formation of cell walls will happen. Cytokinesis will happen. Following that, you will get a 7-celled 8 nucleate embryo sac, which is called the mature embryo sac. The same thing is illustrated here. This is the NCERT diagram. Now it's important for us to understand what are the different cells that are present in the embryo sac. Let's understand that. So this is your functional final embryo sac. At the chalazal end, you will have a total of 3 antipodal cells, all of which are haploid. Remember, every one of these cells, every one of these nuclei will be haploid because it is forming from a haploid megaspore, right? All of them are haploid. These 3 antipodals will be haploid. Towards the micropylar end specifically, you can find 3 cells, 1 egg cell and 2 synergids. Egg cell is towards the center. On either side of the egg cell, you will have synergids. Together, the egg cell and the synergids is called the egg apparatus. Always remember that this must be present at the micropylar end itself. This cell at the center, which I am outlining right now, is large. And at the center, that is why it is called the central cell. Central cell is 1 in number. Within the central cell, there are 2 nuclei. One nuclei moved from the chalazal pole, one that moved from the micropylar pole. Since they came from opposite poles, they are called the polar nuclei. The egg here is haploid. The synergids are haploid. The polar nuclei are also haploid. There are 2 nuclei there. That does not make the central cell diploid. The central cell is multinucleate. It has 2 nuclei, and these nuclei are haploid each. Okay. So totally you have 1, 2, 3 antipodals, 1 egg cell 4, 2 synergids 6, 1 central cell 7, totally 7 cells. 1, 2, 3, 4, 5, 6. Egg apparatus and antipodals have 1 nucleus each. Central cell has 2 nuclei. So totally it has 8 nuclei. There's one interesting feature that we should know about synergids. Towards the micropylar pole of the synergid. See, this is the synergid cell. This is the part of the cell towards the chalazal end. This is the part of the cell towards the micropylar end. Now, towards the micropylar end, you can see that the cell wall has many cellulosic thickenings, differential thickenings. They are called filiform apparatus, and this is a characteristic feature of synergids. You don't find that in other cells. The reason why they are present is because once pollination happens, the pollen tube has to enter the embryo sac. This kind of guides the pollen tube inside the embryo sac and helps it release the male gametes to bring about fertilization. Okay. So this is your functional mature female gametophyte. The reason we are calling this the gametophyte is because it is within this structure that the female gamete is produced. Now, you know that We learnt about the female. Where did that slide go? Yeah. Gametophyte formation. We also saw how within the female gametophyte, the female gamete formed. Now we have to make sure that the male gamete comes in contact with the female gamete so that zygote formation happens. So right now, after the spore formed, everything is in its haploid phase. Once the female gamete haploid, male gamete haploid fuse together, they will form this zygote which is diploid, which will restore the sporophytic phase. Okay. Now, let's see how the male gamete and the female gamete fuses, what all drama happens. Before that, here's a slide that I've added that differentiates microsporogenesis and megasporogenesis. All of the details about the difference between microsporogenesis and megasporogenesis is mentioned here. You can go through it in your notes and you can study from there. It's very easy to understand. All right. Now, let's talk about how pollen grains carrying the male gamete can reach the female gamete that is present inside the ovule. So what happens is, if this is the anther, this anther after it matures will dehisce. It will release the pollen grains that it has produced outside. Okay. These pollen grains will eventually have to get deposited on the stigma. Once the pollen grains get deposited on the stigma, they will form a pollen tube. Inside the ovary, there will be an ovule. Inside the ovule, there is the egg cell, right? So this pollen grain will form a pollen tube. The pollen tube has to enter the ovule, and via the pollen tube, the two male gametes will enter the ovule and the embryo sac and further bring about fertilization. For that to happen, the pollen grains that are released by the anther will have to go and land on the stigma. The process by which pollen grains released from the anther are transferred to the stigmatic surface is called pollination. Now, pollination can be classified into many types based on the source of pollen. It is of three types: autogamy, catenogamy, xenogamy. In autogamy, you see self-pollination happening. Here, you have only one plant involved and only one flower involved. The pollen produced by this flower will get deposited on the stigma of the same flower. Then you call it autogamy. If it gets deposited on the same flower, then you call it autogamy. Auto means self. Here only one plant is involved and only one flower is involved. In catenogamy, only one plant is involved, but pollination happens between two flowers. I'll show you how. Two flowers belonging to the same plant. So this is one flower. This is another flower, both belonging to the same plant, as you can see. Over here, the pollen grains produced by one flower will land on the stigma of another flower. Okay. So the pollen grains that are produced by this, if it lands on the stigma of this, then it is called catenogamy. Here there is one plant only involved and two flowers of the same plant are involved. Coming to xenogamy. In xenogamy, you have two plants involved. So here there are two flowers. The pollen produced by the anther of one flower will get deposited on the stigma of another flower belonging to a different plant. Here, two plants and two flowers are involved. For autogamy to happen, pollination to be brought about within the same flower, the flower has to be bisexual. It is 100% has to be bisexual. Write it down here. The flower has to be bisexual only. If it is not bisexual, self-pollination cannot happen. But in these two cases, the flowers may either be bisexual or unisexual. Okay. Now, in gatonogamy, you are seeing that pollen grains are being transferred between two flowers. So, if you think about it, functionally, it is cross-pollination because it's happening between two different flowers. But these two flowers belong to the same plant. So, they have the same genes as this plant. So, genetically, if you think about it, it is very, very similar to self-pollination itself. When you talk about xenogamy, this is proper cross-pollination because pollen grains from a genetically different plant is brought to the stigma of this plant. This may, if assuming they are not clones, they are not vegetatively propagated, you can say that here the pollen grain is carrying the genotype of this plant. It is coming and pollinating this plant which has a different genotype possibly. So this only is functionally genetically cross-pollination because it brings pollen grains of different types. Okay. So these are the three different types of pollination. Now in self-pollination, for self-pollination to take place, like I said, the flowers have to be bisexual. Is that mentioned anywhere? Okay, not mentioned. And the pollination happens within the same flower. For this to happen, the position of the stigma and the anther has to be very close. They should lie close to each other. And also, at the time when the pollen grains are released, the stigma should have become receptive. Okay? So there should be synchrony in pollen release and stigma receptivity. Synchrony meaning it should happen simultaneously at the same time. Now, there are some species of plants like Viola, Oxalis, Camelina which produce two different types of flowers. One kind of flowers does not bloom at all, it does not open at all, it is always like a bud. Okay. And there's another type of flowers which will open. The kind of flowers which don't open at all are called cleistogamous flowers. These are forever closed. Another type of flowers called chasmogamous or chasmogamous flowers. They are usual, normal, open flowers with exposed anthers and stigma. Okay. There is one advantage of these cleistogamous flowers. You can see here, these are open flowers, chasmogamous, not cleistogamous. There's a mistake here. Let me correct it quickly. These are chasmogamous flowers that are open. In the same plant, in the same species, you can find underground flowers also that don't open at all. They don't even look like flowers, right? They are covered, they are not, colorful. So these are called cleistogamous flowers or open flowers. Now the advantage of having cleistogamous flowers along with chasmogamous flowers is, for cleistogamous flowers to get pollinated, you don't require any external pollinating agent. You don't require wind to come and pollinate it. You don't require any insect or bird to come and pollinate it. Within these closed flowers, obviously the anther and the stigma are going to lie very close to each other. If they are not going to open at all, there is no chance they can get cross-pollinated. The pollen from the same flower has to land on the stigma on the same flower. So 100% invariably they are going to be autogamous or they will always undergo self-pollination. So even though there is no pollinating agent in the form of wind or insects or birds or any other animals, still fertilization will happen. If pollination happens, 100% fertilization will happen. If fertilization happens, seed formation will 100% happen. So in these cases, you can see assured seed set. Assured seed set means 100% guarantee that seed formation will happen because you're not relying on any external pollinating agents. Okay. Now, gatonogamy, I already told you. Here, pollination happens between flowers belonging to the same plant. Though this is functionally cross-pollination, genetically it is similar to autogamy itself. Xenogamy is pollination between two different plants. This is the only type of cross-pollination where genetically different types of pollen grains are brought to the stigma. So in one of these three ways, pollen will reach the stigma of the flower. Now, we have to learn about how are pollen grains carried from the anther to the stigma. The agents that carry pollen grains from the anther when they are released to the stigma are called pollinating agents. Pollinating agents could be living or they could be non-living. If they are living, obviously we call it biotic agents. If they are non-living, we call it abiotic agents. Biotic agents include animals like birds and certain reptiles, insects and all of that. Abiotic agents, you have wind and water. Compared to biotic and abiotic agents, in most cases, pollination happens by biotic agents itself. Abiotic agents is slightly rare. Okay, among wind and water, again, wind is more prevalent, but water is slightly less, very rare. Okay, so be it wind pollination or water pollination or any type of pollination, similar to how you see in animals and humans, the number of sperms that are produced are very high, but the number of eggs that are produced is limited. So same way here, the number of pollen grains are produced in, very, very large quantities, because just because pollen grain is produced and released does not mean it will go and land on the stigma belonging to the same species, right? It could end up anywhere. So in order to increase the chances of pollination, effective pollination, the number of pollen grains are released in very, very high quantities. Okay. Now, what are some characteristic features that we will observe in plants and flowers which are going to get pollinated by wind? Wind pollination is also called anemophily. If a plant or a flower is going to rely on wind for pollination, obviously wind is abiotic. It cannot see, right? So it doesn't matter if the flower is colorful, if it is fragrant, if it is producing nectar or not, because it doesn't matter for the wind because it's not a living thing. So the flowers in this case will usually not spend too much energy in beautifying themselves and putting on makeup, right? The flowers are usually very pale in this case. Tiny, tiny flowers, they will come together in a compact inflorescence because color, what it looks like, what it smells like, what it is going to reward, none of that matters in this case. So here, the flowers are usually packed into an inflorescence. How should the pollen grains be if it has to be carried by wind? It has to be very, very light. Firstly, it's produced in large numbers. Its density is very low because if it's very dense, then because of gravity, it will fall off right where it is released. So it's very, very light, dust-like. So it is, very, very small in size and very light also, not heavy. And another thing is it is non-sticky in nature. If it is sticky in nature, it will go and stick onto random surfaces. We don't want that. We want it to be carried by wind. So it is light, non-sticky, produced in large numbers. What about the stamen? If this is the flower, let's say, it's usually colorless in this case. If this is the petals or some tepals. If the stamen is hiding inside the petals, then if it releases the pollen grains, all of that will fall right inside the flower and it will not effectively be able to cross-pollinate. So the stamens in this case is exposed, right? That means it is exposed, it comes out of the petals or tepals or whatever that particular plant has. So it is very well exposed so that when the pollen grains are released, the pollen grains can be easily carried by wind. How should the stigma be in this case? I'm telling you that the pollen grains are super tiny, very microscopic, very light, small in size. So you can imagine that they are like dust. How do you trap dust? In your house, if there is any small crevices that you want to clean, how do you clean such spaces? You clean such spaces using bristle-like structures, right? Using the, what do you call it? Mm, to clean dust and spider webs and all of that, you use that bristle-like structure, right? Feathery structure, because that will trap dust very easily. If you try to clean such surfaces where small dust particles are there with a cloth or something with a greater surface area, then it will not get cleaned very well. Same thing happens here. In order to trap those tiny, tiny particles that are moving away with wind, if there is a broad knob-like stigma, it will not be able to capture the pollen grains very effectively. So the pollen grains here are large and they are feathery in nature. Because of its feathery nature, it can easily trap the pollen grains that are being carried by wind. So the stamens are very well exposed. The stigma is large and often feathery so that it can easily trap the pollen grains. Now in such flowers, every ovary will have only one ovule. Such a type of pollination is common in grasses. I'm sure you've seen the inflorescence of rice and wheat and maize and ragi and all of those. In those, you don't see very bright, colorful flowers. It's not even like the fruits are produced separately. All the fruits are produced inside the same inflorescence, right? Because all the flowers are packed within the same inflorescence. You may have seen the silk-like hair arising from the corn. They are nothing but the stigma and style of that flower that were meant to trap the pollen grains. Okay. Now, how about water pollination? Water pollination is common in few aquatic plants which are submerged. Aquatic plants are plants that grow in water. Even though aquatic plants grow in water, not all aquatic plants rely on water for pollination. Only if the flower is produced inside the water will pollination happen by water. Lotus is an aquatic plant, water lily is an aquatic plant, but over there pollination is not done by water because the flowers are produced at the surface, right? That's the reason why those flowers are colorful, because probably they are pollinated by insects and they want to attract insects. If it is pollinated by water, then there is no need for those flowers to also spend energy in becoming colorful, because water again is abiotic, it does not see, right? It cannot see how beautiful the flower is, it will not be attracted to it, it's just randomly, along with its movement, it will passively carry the pollen grains. Not all aquatic plants are pollinated by water, keep that in mind. In plants where flowers are produced at the surface, like in lotus and water lily, either wind or insects only will pollinate it. Mostly it is insects. Okay. Now, pollination by water is very, very rare. In all of angiosperms, you can mostly see it in monocots, and that also is restricted to just about 30 genera. Okay. So, you have two examples to learn here. One is how pollination happens in Vallisneria, which is a freshwater aquatic plant that is submerged. And another one is how it happens in Zostera, which is also called seagrass. Now, let's see how it happens in Vallisneria. Hydrilla also is a freshwater plant which undergoes pollination by water itself. You have to learn about Vallisneria and Zostera specifically. So, in Vallisneria, it is a dioecious species. There is a separate male plant, separate female plant. The female plant produces flowers. Okay, so the flowers will have a coiled pedicel like this. They will be submerged inside water. Okay, so think that all of this is water. Okay, so there's water completely here. These are submerged inside water. This is the female flower. When the female flower matures, when the stigma becomes receptive, this will get uncoiled and the flower, same flower, will reach the surface of the water body. Water surface it will reach. The male plant will produce the male flowers. The male flowers are directly released into the water. The male flowers will start floating in water here. They can't actively swim. Along with the water currents, they will passively float. If by chance they encounter a female flower, they will bring about pollination. After fertilization happens, this will coil back and get inside, and the fruit will eventually form inside water itself. And after the fruit is formed, the seeds will be released inside water. The seeds will germinate inside water and give rise to new plants. So here pollination happens at the surface level. This is called epihydrophily. Water pollination is called hydrophily. So here epi means above. At the surface level it is happening. That's why we call it epihydrophily. The exact same thing is mentioned in the text here. You can refer that in your notes. Now, in sea grasses like Zostera, what happens? Let's see. Here hypohydrophily will happen. In Zostera also separate male flowers, separate female flowers are produced. It is a monoecious plant but it produces unisexual flowers. Separate male flower, separate female flower. Both of them are submerged inside water only. None of them will come to the surface. The male flower will produce pollen grains and release it into the water. Inside the water itself, submerged condition itself, the pollen grains will start floating, not floating, floating is surface, will start, moving along with the water currents. Its shape will help it. Unlike pollen grains that are pollinated by wind or insects, the pollen grains here are long and ribbon shaped. Because of its shape, it can passively float or move inside water according to the water currents. If it ends up meeting the female flower, meeting the stigma, it will bring about pollination there. Since here pollination happens within the water body itself, not at the surface, it is called hypohydrophily. Hypo, I'm sure you know, means underneath or below, right? So that is hypohydrophily. Regardless of whether it is epihydrophily or hypohydrophily, generally pollen grains that are pollinated by water will have a mucilaginous coating outside, a jelly-like coating outside that prevents it from wetting. Remember that. Okay. Now, that's about water pollination. Pollination by animals is called zoophily, that is biotic pollinating agent. You have many animals that are pollinators such as birds, beetles, butterflies, bees, flies, wasps, ants, moths, so many types of insects. Some rodents as well, some reptiles as well are known to bring about pollination. But among them all, insects are the major pollinating agents, and among insects, bees are the most dominant type of pollinating agent. Among abiotic is more predominant. Among abiotic, wind is more predominant. Among biotic, insects are more predominant. Within insects, bees are most dominant type of pollinating agents. Remember that. Okay. Now, what are some of the characteristic features of flowers in which insect pollination will take place? See, it's important for flowers to bring insects to them, draw the attention of insects to them. So they have to be attractive because insects are animals that have sensory structures. They have eyes that can see. They have olfactory sense that can smell. They eat food. So if there is something tasty, they can go pick it up from there, right? So in order to first attract the insects, they have to appear attractive. For that, the flowers in this case are usually very brightly colored and large in size. If the flowers are small in size, they can't catch the attention of the insects. So they will, aggregate to form an inflorescence so that it will appear larger and grab the attention of the insect. So the flowers here are generally large and colorful so that it can attract the insect because of how it looks. Another way in which flowers attract insects is because of its smell. Okay. Most flowers produce fragrance, like it smells good. However, there are some flowers that also produce foul odors, bad smell. Such flowers attract pollinators in the form of flies. You know how right around cow dung there will be flies present, right? Such flies are attracted to the smell of cow dung. There are literally flowers that smell like cow dung because flies such as those dung flies and beetles will be attracted to it, and then it will bring about pollination. So the idea of these flowers is that pollination has to happen. How it happens doesn't matter. They don't care about whether they are smelling good to us or not. They want pollination to get done. So whatever helps in them getting pollinated, they will do all of that. So it could be fragrance or it could be color. Either ways will attract insects or pollinating agents towards them. They are called attractants. Attractants is what will grab the attention of the pollinating agent. Now, okay, a flower was super attractive. It gave out like a great fragrance. So one insect was attracted towards it and it went there. But once it went there, it couldn't find anything. Why would an insect want to go back to a place where it's not getting anything? right? In order to make sure the insects keep on visiting these flowers again and again, the flowers should give away some rewards. Okay, if a insect goes there and gets some reward, it'll be happy and it'll feel like going there again. So in order to make sure that the insects keep visiting them again and again, the flowers offer some rewards. 3 rewards you can see in flowers. Firstly, it will offer it nectar. Nectar is the juice that insects will drink. It is nutritious. The flowers produce them. It's sweet, sugary kind of juice that insects can drink and gain nourishment from it. Secondly, it offers snacks in the form of pollen grains. So the pollen grains that are produced by the anther, some of it is used as a reward for the insects to bring about pollination. Thirdly, in some species only, the flowers also provide space for these insects to lay their eggs. Not just lay their eggs, even after the eggs are laid, while the larvae are getting produced, while the larvae are developing, the plant will offer some of its own seeds for the larva as food, so that these insects will keep on visiting the flower and bring about pollination. So it's like a mutualistic relationship between an animal and a plant, where both of them are getting benefited because of this interaction. Okay, so here usually they are large and colorful, and they will give out fragrance that may be, you know, good smell, or it may secrete foul odor. If it secretes foul odors, it will attract certain flies and beetles. So these are basically attractants, and as rewards they will offer nectar and pollen grains, and sometimes they will also provide a safe space for the insects to lay eggs. Such a thing you can find in Amorphophallus, which is the tallest flower of an angiosperm, and you can also find that in another species of plant called yucca. This will offer space for the yucca moth to lay its eggs inside. And sometimes it so happens that insects don't know that they're bringing about pollination. They have no idea that they're doing something so priceless, such a valuable service in nature. They go there for nectar and pollen grains. Sometimes it so happens that they will go there, pick up pollen grains and drink nectar from there and leave without bringing about pollination. So in that case, you call such insects as pollen or nectar robbers. Actually, plants have to evolve in order to minimize um, such things. Okay, so that's about pollen and nectar robbers. What do we have next? See, pollination is of many types. Self-pollination can happen, cross-pollination happens. Self-pollination has its own advantages. If self-pollination happens, there is no reliance on any other kind of pollinating agent. After pollination happens, seed set will be there, ensured guaranteed seed formation, guaranteed next generation will form. But sometimes after self-pollination, repeated self-pollination, what happens is the offspring that are produced, the plants that are produced will not be strong enough, they will not be hardy enough, they will not have a lot of resistance against changing environmental conditions, they will not have disease resistance, they will be very weak, and if they are weak like that, they will not be able to reproduce further and continue the generation. That species will cease to exist if that happens. Plants know this. Plants are very clever. They are brainless organisms, but they are super clever. So plants know that if repeatedly self-pollination happens, the further generations will not be healthy. Okay. Ultimately, the biological purpose of all living organisms on this planet is to make sure that the species continues. Plants also want that. They want to make sure that their babies and the next generation are healthier and make sure that the species will continue. So in order to prevent that phenomenon where the offspring are formed weaker, they will come up with devices. They have evolved and come up with devices which will make sure, or which will kind of encourage cross-pollination and discourage self-pollination. Repeated self-pollination will cause what is called inbreeding depression. Inbreeding depression is just what I mentioned. Like they will be very weak and they will not be able to grow well, develop well, and have their own offspring, next generation. So that is inbreeding depression. We see that in humans as well. Whenever very close relatives, get married, usually the chances of their children having genetic disorders is very high. So they could be deaf, they could be mute, they could have mental retardation. We've seen this. You may have seen this in your circle. So that can happen even in plants. In plants, what does close relatives mean? It's like self-pollination. Within the same plant, the same genotype again and again getting pollinated. inbreeding depression will happen. So in order to avoid inbreeding depression and make sure that the next generations are healthier and they will continue to contribute to, continuation of the species, they have come up with these devices that will discourage self-pollination and encourage cross-pollination or outbreeding. And such devices are called outbreeding devices. Okay. Now, there are 4 outbreeding devices that you must learn about. The first one is, for self-pollination to happen, the flower has to be bisexual. Not just that, the pollen grain, whenever it is released, the stigma of the same flower should be receptive at that time. Okay. Stigma being receptive means it should recognize, okay, there's a pollen that has fallen on top of me. Does this belong to the same species? Does it have the same genotype? It has to check all that, and only then it will allow the pollen tube to form. If the stigma is not yet receptive, it's not yet mature, it will not allow formation of pollen tube. Okay. So at the same time that the pollen grain is released, the stigma also has to be receptive. Only if both are happening simultaneously, there is synchronous pollen release and stigma receptivity, only then self-pollination can happen. To prevent that, there is, either pollen grains are produced first while the stigma is not mature, or the stigma becomes receptive first. At that time, the pollen grain would not have gotten produced. So, there is no synchronicity between the release of pollen grains and the stigma receptivity. It is not synchronized. That means they don't happen at the same time. When the pollen is released, even if it falls on the stigma of the same flower, the stigma will not be receptive. Or when the stigma is receptive, the pollen grain is still developing, it is not yet released. That way, even though a flower is bisexual, you can prevent self-pollination. That is one way. Another way is physically separating the stigma and pollen anther within the flower in such a way that even if the pollen grains of the same flower are released, the chances of it going and getting deposited on the stigma of the same flower is very, very minimal. Okay. So there is difference in the positions of anther and stigma so that pollination within the same flower will not happen. What do we have next? Self-incompatibility. Self-incompatibility is like a genetic mechanism. It's like Tinder for plants. So the stigma here is receptive. It will receive pollen grains from same flower, same plant, different plant, all of that. It will check for the genotype. Okay. So if it is from the same species, okay. If it is from the same species and the same plant, it's like, no bro, I'm not okay with this, and it'll swipe left. If it is from a different species carrying a different genotype, only then will it accept the pollen grain and allow the formation of pollen tube. Otherwise, it will not allow pollen germination at all. For example, you see here, this is one plant that has its genotype S1, S2. Okay, so this is the kind of genes that this plant has. If S1 genotype carrying pollen grain comes and gets deposited on the stigma, pollen tube formation is not allowed. If S2 comes and forms on top of it, pollen tube formation is not allowed. Only when pollen grains carrying different genotype like S3 and S4 for instance, comes and gets deposited on top of the stigma, only then will pollen tube formation be allowed. Okay, so that is called self-incompatibility. Now lastly, produce unisexual flowers. If you produce unisexual flowers, then obviously autogamy cannot happen. Okay. Now, the plant could be monoecious. You have one plant. Okay. Within this one plant itself, you have both types of flowers. You have the male flower having only the anther. It does not have the pistil. It is a male flower. And you have another flower that has only the stigma. Since it has only stigma, there is no way self-pollination or autogamy can happen. Here it has only anther, so there is no way autogamy can happen. In this case, the pollen grains released by this plant, this flower, can go and get deposited on the stigma of another flower. When we say it is monoecious, it means there is only one plant. When we say it is unisexual, that means flowers are of two types, separate male, separate female. Unisexual is the term used to describe a flower. Monoecious is a term used to describe the plant. When we say the plant is monoecious, that means both male and female parts are present on the same plant. It need not necessarily be present in the same flower, but it will be present in the same plant. If it is present within the same flower, you call it monoecious bisexual. Here it is monoecious unisexual. Each flower has only one reproductive part. So in this case, geitonogamy can happen, but autogamy cannot happen. For autogamy to happen, flowers have to be bisexual. As opposed to this, you have dioecious plants where the plant itself is separate. There is a separate male plant and a separate female plant. The male plant produces only the male flowers carrying or bearing the stamens. The female plant will produce flowers that only bear the pistil. In such cases, autogamy also cannot happen, geitonogamy also cannot happen, only xenogamy can happen. So this will prevent autogamy as well as geitonogamy. This is the ultimate way to prevent self-pollination. Okay, so here 100% of the time cross-pollination itself will happen. Example, in papaya you can find separate male plant, separate female plant. Male plant will produce only male flowers with stamen only. Female plant will produce only female flowers with pistil only. So in that case, you can ensure that always outbreeding or xenogamy itself will happen. Okay, so that's about outbreeding devices. Now, the pollen grain has been released by the anther. By one means or the other, the pollen grain would have landed on the stigma. So let's see what happens at the stigma. The stigma is the receptive surface for pollen grain. So this is the stigma, this is the style. Let's say this is the ovule. There will be many ovules, but inside the ovule, the embryo sac is here, and inside the embryo sac, the egg cell is there. This pollen grain is now carrying two male gametes inside. Now, once the pollen lands on the stigma like this, the chemicals present on the exine of the pollen grain will interact with the chemicals present on the stigmatic surface. You know, in oceans and in water bodies, there are so many different species of fishes, and all of them will undergo external fertilization. Sperms are released out in the water, eggs are also released in the water. Sperm and egg will fuse in water itself. A shark would have released its sperms and eggs, and so would have a clownfish. But you know, a shark's sperm cannot fertilize a clownfish's egg, or a clownfish's sperm cannot fertilize a shark's egg, because they are species specific. Very similarly, you would have learnt about this in human reproduction as well. There is that calcium ion reaction that happens when a sperm fuses with the egg, and there are so many receptors that are present in the egg surface that can specifically bind to the sperm only if it is belonging to the same species. Otherwise, that interaction at the surface cannot even happen, and the sperm nucleus cannot even enter the egg nucleus. Egg cell, right? So something similar happens in plants also. In nature, you can find so many different species of plants. So many of them produce flowers. If they're producing pollen grains and releasing them at the same time, you can't ensure that this species' pollen grain is going to fall on this species' stigma itself. It also can happen that some other species pollen grain can land on the stigma. You do not want to waste your energy in allowing pollen grains of random species to germinate on your stigma, right? So there has to be some way, there has to be some checkpoint where there's somebody like literally guarding and checking whether the pollen grain is of the same species, whether it is compatible or not. The stigma does that beautifully. So there is communication that happens between the surface of the pollen grain and the surface of stigma. They do not talk in English and Kannada and Hindi like us. Instead, they communicate by means of chemicals, that is called a chemical dialogue. So there are exchange of molecules that happens, and by means of recognizing the molecules present on the surfaces, the stigma will either accept the pollen grain or reject the pollen grain, similar to your Tinder left swipe and right swipe. Okay. For the stigma to accept the pollen grain, it has to be compatible. That means 100% it has to be belonging to the same species. And if it is a type of plant, a species which has genetic incompatibility, like if it has self-incompatibility, then it also has to make sure that it does not carry the same genotype as the parent plant, as the sporophyte of that plant. So once it checks both boxes, it knows that it is compatible type, only then will it allow the pollen grain to form the pollen tube. When the pollen grain forms the pollen tube, you know how inside the pollen grain there is the wall called the intine, and outside there is the exine. Inside the pollen grain you have three cells, a vegetative cell and two male gametes in the mature pollen grain. So basically by means of the germ pore, the pollen tube will form here. When we say pollen germinated, it means the pollen grain formed a pollen tube inside the style. Okay. So this vegetative cell itself will form the pollen tube. That is why the vegetative cell is also sometimes referred to as the tube cell. Now for the pollen tube to grow deeper inside, nourishment is required. That is why the vegetative cell will have abundant nutrient reserve. Along with that, the stigmatic surface also will contain certain sugars which will nourish the formation and elongation of the pollen tube. The style also will contribute to that, and through the style, the pollen tube will form. Ultimately, the pollen tube will get deeper and deeper and deeper, and it will enter the ovule, and inside the ovule it will enter the embryo sac, and once it's inside the embryo sac, it will release both of the male gametes. Okay. So all of these processes that take place, all of the events that take place from the time the pollen has landed on the stigma to the time the pollen enters the ovule, all of them come under pollen-pistil interaction. Okay. So here, it's not necessary that for one stigma, only one pollen grain will form. In nature, many tiny pollen grains can come and get deposited on the stigma. Depending upon whether it is compatible or not, the stigma will decide. Okay, so if it is compatible, it will allow the formation of pollen tube. If it is incompatible, pollen tube will not form. At the same time, many pollen grains can form pollen tube and enter the ovule. Many ovaries have many seeds, so each pollen tube will enter different ovules and bring about fertilization. Okay, so here you can see this is an image taken from under the microscope where this is the style, this is the stigmatic surface. You can see so many pollen grains landing on top of it and also forming pollen tubes, right? Yeah, so that is pollen-pistil interaction. Ultimately, what happens? The pollen tube will enter the ovule through the micropyle. Okay, so let's say this is the micropylar pole. Once it enters the micropyle, what do we have at the micropyle? You have two synergids and one egg cell. Here, this is one synergid with its filiform apparatus here. This is one egg cell. Another synergid is hiding behind. You can't see it from this angle. So the pollen tube will not enter directly the egg cell. It will enter one of the two synergids. The synergids will guide the pollen tube. Once it has entered the synergid, the pollen tube will break open, releasing both of its gametes. It was carrying two male gametes, right? So let's say this is one male gamete, this is one male gamete. Two of them it was carrying. It will release the two male gametes inside. That is what will happen. Now, after the two male gametes have been released, one male gamete will fuse with the egg cell and result in the formation of zygote. The other male gamete, the second male gamete, okay, just give me. Yeah. The other male gamete here will fuse with the two polar nuclei to form a structure called primary endosperm nucleus. By the time the second male gamete fuses with the two polar nuclei would have themselves fused in some cases to form what is called the secondary nucleus. The secondary nucleus will be diploid. If it is two polar nuclei separately, then it is haploid and haploid. So, two fusion events will take place in angiosperms. Because two fusion events are taking place, we call this double fertilization, and this double fertilization is a characteristic feature of angiosperms. You don't find this in any other plant groups. So, the first fusion event is when The male gamete that is haploid fuses with the female gamete or the egg cell, which is also haploid, and gives rise to the formation of a diploid zygote. You know, usually fertilization has another name. It's also called syngamy. The other male gamete, which is haploid, will fuse with the two polar nuclei, which are haploid each, or it will fuse with the secondary nucleus, which is diploid, to form a triploid structure called primary endosperm nucleus. The cell within which the primary endosperm nucleus formed was earlier called central cell. Now that cell will be called primary endosperm cell. So that is a single cell carrying a triploid nucleus. The zygote is one single cell having a diploid nucleus. Now eventually these two cells will undergo cell division to give rise to multicellular structures. The zygote will undergo mitotic divisions to give rise to the embryo, which is the future plant. The primary endosperm nucleus again will also undergo mitosis to give rise to the endosperm. The endosperm's function is to nourish the developing embryo. When double fertilization happens, double means two times, two fusion events are happening. Syngamy and triple fusion, both of them will happen almost simultaneously, almost at the same time. Okay. So, the zygote and the primary endosperm nucleus are almost formed simultaneously. But the development of zygote into the embryo will happen slightly slowly compared to the development of primary endosperm into the endosperm. This will happen slightly faster in comparison. So we say that the formation of endosperm precedes the development of embryo. Though zygote and PEN have formed simultaneously, endosperm development will happen sooner than embryo development. There is a reason for that. For the embryo to develop, for the zygote to develop into the embryo, mitotic cell divisions will take place, and that requires energy. And for that energy, nourishment is obtained from the endosperm. In order to make sure this development happens, some amount of endosperm has to be formed. Without this food, this development cannot happen. So this happens first. Once there is sufficient food reserve, once there is sufficient nourishment, then this will start utilizing this and dividing to give rise to the embryo. Okay. So that is why the endosperm development happens sooner than the embryo development. Now we have to talk about endosperm and embryo. Endosperm is formed from the primary endosperm cell, which is triploid. This primary endosperm cell has the nucleus, which is called primary endosperm nucleus. This will undergo a series of mitotic divisions. When it undergoes mitosis, again similar to how embryo sac development happened, karyokinesis will happen. Karyokinesis will happen, but cytokinesis will not immediately happen. As a result, nuclear divisions will happen, but there are no cross walls laid between the nucleus. Such an endosperm is called a free nuclear endosperm. Eventually, as it develops further, you can see that cytokinesis will happen and wall formation will happen. This is called cellular endosperm. Every one of the nuclei in the endosperm will be triploid here, because it is formed by mitosis of a triploid cell. For example, if you see tender coconut water, that we use, that we drink, the tender coconut water, is actually free nuclear endosperm. Once cell wall forms, the kernel of coconut will form. The part that is hard inside, the white color part, that is an example for cellular endosperm. If this is like the tender coconut, okay, so inside that whatever water will be there, the tender coconut water that you see, that is free nuclear endosperm. If you open a coconut, if you cut open a coconut, when you cut open a coconut, like it has all of this fiber and everything inside, when you cut it open, you see this white color kernel inside. This white kernel that you see inside a coconut, that is called, that is the cellular endosperm. Okay, so this whatever you drink here, that is your free nuclear endosperm. Okay, now endosperm, like I mentioned, is a triploid tissue. It is filled with reserve food. It is used for the nourishment of the developing embryo. In most common type of endosperm development, first free nuclear divisions will happen, that's called free nuclear endosperm. Later cell wall formation will happen, that is called cellular endosperm. So for example, coconut water is free nuclear, kernel of coconut is cellular endosperm. There are many species of plants in which by the time the seed matures, endosperm will be fully consumed. Endosperm will not exist in the mature seed. However, there are certain species where endosperm will exist even in mature seeds. Okay, so that difference you can see. More about which are those seeds and examples we will learn as we move on. Okay, for now, okay, here there are examples that I've given here also. Endosperms is fully utilized in peas, groundnut and beans, mostly in dicot plants. It will persist even in the mature seed in castor and coconut, mostly monocots, but castor is a dicot. Okay, so basically what happens is endosperm is like food. Embryo is using that food to grow. As embryo grows, it will use the food. So as development of embryo happens, the quantity of embryo, the number of cells in the embryo will increase, but the quantity of endosperm will keep on getting reduced. In some species, the endosperm is fully utilized for the embryo development. So in the mature seed, endosperm will not persist at all. It will not exist at all. In some cases, some amount of it will still be present. Remember, all of this is happening inside the ovule. Where did this whole drama happen? All this embryo sac, double fertilization, all of that happened inside the ovule itself. So that ovule itself is transforming into the seed. Within the ovule only, zygote formed. Within the ovule only, endosperm formed. Both of them are present inside the ovule. As the zygote is developing into the embryo, the ovule will get transformed into the seed. So it is within the seed that the embryo will be present. It is within the seed that the endosperm also will be present. Okay. So at maturity, the seed may or may not have endosperm. In some species, endosperm will still be present, and some it will not be present. Okay. Now, let's talk about embryo. Embryo develops from the zygote by mitosis. Zygote is a single cell, diploid cell that will undergo mitotic divisions to give rise to 2 cells, 4 cells, and eventually it will start dividing and differentiating and give rise to the embryo. In embryos, you have many stages. In monocots and dicots, embryonic development, there are differences, but the initial few stages are very similar. The embryo first develops, the first embryo that develops is called the pro-embryo stage. Eventually, it will form a globular embryo. The reason we are calling this globular is because you can see like a spherical part here. This is globular, globe-like spherical. That is why it's called globular embryo. Over here, there is this long, slender filament sort of a thing, multicellular. This is called the suspensor. Okay. This is called the embryo proper. It is this embryo proper which will give rise to the future plant. When you sow a seed in the soil, the seed will germinate and give rise to a new plant, right? That plant will come from this part of the embryo itself. What is the function of the suspensor then? The suspensor is meant to penetrate deep inside the endosperm tissue and draw nourishment from it. So if this is the endosperm tissue, inside the seed. Let's say this is the seed, this is the endosperm, and the embryo is developing here. Okay, so this is the embryo, this is the suspensor. From here it will draw nourishment and pass it on to the embryo so that it develops further. Imagine it in 3D. Okay, so there's a tissue. Inside that tissue, the suspensor has penetrated. So that's the job of the suspensor. Eventually, in dicot plants usually, this proembryo will form a heart-shaped structure that is called heart-shaped embryo, and eventually it will mature into the mature embryo. Each one of these lobes will become the 2 cotyledons of dicot plants. That's how it is. It's important for you to remember the exact sequence. First it is globular, then it is heart-shaped, then it is mature embryo. The function of the suspensor here and anywhere in an embryo is to penetrate into the endosperm tissue and draw nourishment from there. Okay. Now, we have to learn about the structure of dicot embryo as well as monocot embryo. Let's quickly do that. Remember that the embryo is what is going to give rise to the future plant. So the embryo is going to have an axis similar to the stem. And this is called the embryonal axis. Similar to how you have nodes on the stem, you have node on this axis as well. And at this node, the 2 cotyledons will be attached. This node is called the cotyledonary node. Since this is dicot embryo, these two are the 2 cotyledons. The part of the axis that lies above the point of attachment of cotyledons here, this is called the epicotyle. Epi means above. The axis that is present above the point of attachment, above this point. The part of the axis present below the point of attachment, this is called the hypocotyle. Now the tip of the epicotyle is called the plumule. The plumule is what will give rise to the shoot system in the plant. At the tip of the hypocotyle, you have what is called the radicle, which will eventually give rise to the root system of the plant. Remember, this embryo lies inside the seed. Okay. So within the seed, embryo is there, and when the seed germinates, it is this embryo itself which will give rise to the future plant. So the plumule will contribute to the shoot system and the radicle will contribute to the root system. This is what will happen. But now, in order for you to understand the different parts, I have kind of elongated this axis and shown you what it is. But in an actual seed, if you have seen the seed of peanut or green gram, you will know that when you open the 2 cotyledons, there will be one very small stick sort of thing there. That is how short the axis will be, but for you to understand the different parts, I have drawn it much elongated. It will actually be like very small. Okay. Now we look at the same parts over here. This is our embryonal axis. This is the point at which the 2 cotyledons are attached. This is one cotyledon and this here is the other cotyledon. The point of the axis above the point of attachment is the epicotyle and the tip is the plumule. This is the hypocotyle and the tip of the hypocotyle is the radicle. Covering the radicle, you will have the root cap. This is the structure of the dicot embryo. When you talk about the structure of monocot embryo, that is also similar, but in monocots, you have a single cotyledon. If this is the embryonal axis, this large single shield-shaped cotyledon is what we call the scutellum. It has only one cotyledon. There is a small extension sort of a thing, appendage that arises from the cotyledonary node itself, which is called the epiblast. This is like a remnant of the other cotyledon. The other cotyledon is not large, right? So it's reduced like that. Now this is the point of attachment. This is your epicotyle and this is the hypocotyle. At the tip of the epicotyle, you have the shoot apex. At the tip of the hypocotyle, you will have the radicle. Covering the radicle, you will have the root cap present. But if you notice, there are 2 additional terms mentioned here. It is coleoptile and coleorhiza. Coleoptile is the sheath-like structure that covers the shoot apex or the shoot tip or the plumule in monocot specifically. This is found, this term is used exclusively in monocot embryo. This is also exclusive to monocots, and so is coleorhiza. Coleorhiza is the sheath-like structure, protective covering that will cover the radicle. Okay, so that is about your monocot embryo. Now moving on, we have to learn about one technique that is used by plant breeders, which is called artificial hybridization. Let's take for example, there are two plants belonging to the same species. They are two different varieties. Okay, so this one plant produces flowers that are large and brightly colored in more quantities. So it gives a high yield. Okay. So both of these plants belong to the same species. They are just different varieties having different characteristics. As opposed to that, this plant belonging to the same species produces flowers that are much smaller in size, produces flowers of a color that is not so much in demand in the market. Okay. So this plant produces high yield. The number of flowers per plant is very high. This plant produces low yield. Now this plant this plant does not have disease resistance. Okay. So it has less disease resistance. Any pest infestation or pathogen infestation, it will immediately die. But this has very high disease, or let's say pest resistance. We call it disease resistance here. So I'll retain the same thing. Disease resistance. Okay. So the plant breeder wants to make sure that he combines the high yield property from this plant along with the disease resistance from this plant into one hybrid plant. He wants both of these features to be combined in one hybrid. So he has to artificially hybridize them, artificially pollinate them. Just by artificially hybridizing them, you cannot ensure that you will get this combination in the hybrid. You might as well less disease resistance and less yield in the hybrid. So later they will have to select by looking at the offspring. Okay, so basically the idea is to bring together this characteristic of this plant and this character of this plant into a common hybrid. So what are the steps that the plant breeder will have to employ in order to artificially hybridize plants? We will see. Firstly, if it is a bisexual plant, you will have to designate one plant as the female plant and another plant as the male plant. Okay, for now, let's say both of these are bisexual plants, and I will consider this to be the female and this to be the male. What are we going to do next? You will take this plant that produces bright pink flowers in large quantities. Okay. This plant will have its own stamen. It will have its own stigma as well. I said, right, they are bisexual. Now this other plant also will have its own stamen and its stigma. Now I am designating this plant as female and this plant as male. If I want to call this plant as female, I have to cut and remove the male part from it, right? If I am calling it female, ideally what do I want from here? The pollen grains that this produces should go and get deposited here. I do not want the pollen grain from the same plant getting deposited here. I do not want some random pollen grain getting deposited here. So in order to make sure that the pollen from the same flower does not pollinate the stigma, what I'm going to do is, even before the anther matures, even before the anther dehisces, I will cut the anther and remove the anther from the flower in its bud condition itself. This process is called emasculation. E means removal, masculine male part of the flower, the stamen that is removed much before the anther dehisces. Okay, that's the first step. Now that will ensure that the pollen from the same flower will not pollinate the stigma. How can I ensure that some random pollen does not come and fall on this? How can I make sure that this pollen only will fall there? I will tie a bag around this flower. And that step is called bagging. The second step is called bagging. What does bagging do? Bagging makes sure that unwanted pollen will not come and contaminate the stigma before it becomes receptive. Once this pollen grain, this flower has produced its pollen grains, I am going to remove the bag, dust specifically this pollen grain on top of the stigma and then cover it with the bag again. The next step is dusting with pollen and then covering it with bag again, which is called rebagging. Once you pollinate it with your choice of pollen grains and cover it again, it is safe from contamination. Fertilization will happen, seed formation will happen, fruit formation will happen. You will allow the fruit to develop inside the bag itself. Later, you will collect the fruit, take out the seeds, sow them in the soil. Look for which of those offspring has your desired combination of characters and select that. That is what people do. Plant breeders. That is called artificial hybridization. You're producing a hybrid artificially depending upon your desired characters. Now the first step here, which is called emasculation, where we removed the anther from the bisexual plant which was designated as male, you won't have to do that if a plant is unisexual. If a plant has only pistil, it does not have anther at all, it does not have stamen at all, if it is a female flower, unisexual flower, there is nothing for you to cut there, right? So if it is unisexual, there is no need for emasculation, you might as well do the other steps, okay? So that's about artificial hybridization. Now we have to talk about the seed and the fruit. Seed is a characteristic feature of sexual reproduction in angiosperms. It is a product of sexual reproduction. When the ovule gets fertilized, ovule transforms into the seed. So the seed can also be called as a fertilized ovule. Where are the seeds formed? It is formed inside the fruit. What are the different parts that you can find in seeds? The two integuments that were there in the ovule will transform into what we call the seed coats. The integuments protected the nucellus and the embryo sac. The seed coats will protect the embryo and the endosperm tissue. Okay, protection is its job too. It will become much drier. And then inside the seed you have the embryo, you also have the endosperm. Endosperm may or may not be present in the mature seed, but embryo will be there, and part of the embryo will be the embryonal axis. Attached to the embryonal axis, you will have one or two cotyledons depending upon whether it is monocot or dicot plant. Usually in dicot plants, you will have cotyledons which store reserve food. Okay. Now, depending upon whether at maturity endosperm is present or not. Remember, endosperm will always get formed. It's not like some seeds endosperm will not form and some it will form. It will form in all of them. But at maturity, if endosperm is still present or not is what we use as the basis for classification here. If in mature seeds endosperm still persists, then we call it endospermic seeds or albuminous seeds. If endosperm is used up during development of embryo and in the mature seed you do not find any embryo left, then you call it a non-endospermic, non-albuminous or exalbuminous seed. Examples for albuminous seed where endosperm is present, usually monocots like wheat, maize, barley. Castor is an exception, it's a dicot plant. Example for non-albuminous seeds where endosperm is completely consumed, dicot plants like ground nut, pea. Okay, in them the food will be stored in the cotyledons. Now, in some plants, in most plants what happens is, once pollination happens, once fertilization happens, the ovule, the integuments will transform into the seed coats. Within this seed itself, you will have the embryo, you will have the endosperm, the nucellus that was present, the nucellar tissue that was present around the embryo sac will kind of disintegrate. Okay, it will degenerate. But in some species like black pepper and beet, nucellus will still persist even in mature seeds. Some amount of nucellus will persist in the periphery of the seed like this, like a layer it will form. So this persistent nucellus that you can find even in mature seeds is what is called perisperm. Peri means around, sperma means seed. In the seed, within the seed, around the embryo, you can find a layer of nucellus still existing at maturity. Then you call that perisperm. Okay. Now, here are some structures of different seeds, monocot seeds, dicot seeds, given at different, uh, you know, angles. So take a look at all of these diagrams. These are diagrams from NCERT itself. Learn all of their labels. I've explained all of them. So just take a look at them. Okay. So some of them are dicot seeds, some of them are monocot seeds. So take a look and learn their labels. It will appear in your NEET exam. It may appear in your NEET exam. Now, seeds offer many advantages to angiosperms. You know how, if you look at the plant kingdom, gymnosperms and angiosperms are the only plant groups that produce seeds. Before that, prior to that, your pteridophytes, your bryophytes or algae, they are not seed producing plants. Seeds offer many advantages to both gymnosperms and angiosperms, but in angiosperms, because surrounding the seeds there is a fruit also, which gymnosperms don't have, there is additional advantage that seeds produce to angiosperms. You know, seeds have a fruit wall covering them, right? Because they have a fruit wall covering them, they have evolved many mechanisms for dispersal of seeds to new habitats. If a tree is here, if a plant is formed seeds here, if it has produced flowers and then the seed has formed, fruit has formed at this point, if you want this plant species to, get distributed and increase their area of distribution geographically, if the same plant wants to get colonized in newer areas, it can happen with seed dispersal mechanisms. Some plants produce fleshy fruits that are juicy and nutritious. Many animals come and feed on them. When animals come and feed on them, like squirrels or some other animals or birds come and feed on them, along with the fruits, they tend to eat the seeds also. So seeds goes into the digestive tract of the animal, and then that animal might move somewhere else and go and defecate there. So along with its fecal matter, the seed is going to go and get lodged in some other brand new area. Now that it is trapped within the fecal matter of that animal, it has got manure also. It has got nutritious organic matter present in the form of fecal matter. So it can germinate there in a new area and survive and establish itself there. There are some plants that don't produce fleshy, juicy fruits. Instead, they produce dry fruits. They also will have mechanisms to disperse their seeds. Like some plants like lady's finger, if you don't pluck them, if you leave it by itself, to itself in its natural habitat, the lady's finger will open itself, the fruit wall will open itself and then release the seeds outside so that it gets dispersed to newer areas. So one way or the other, these angiosperm plants have evolved mechanisms to make sure that the seeds will get dispersed farther and farther away to increase their area of distribution. Okay, now within a seed, either in the form of endosperm or in the form of cotyledons, there are sufficient food reserves. Within a seed, you have a tiny baby embryo present that is going to germinate and give rise to a new plant. Until that new plant forms, produces leaves, starts to photosynthesize, the cells have to survive there. That requires energy, right? Because the seed has abundant reserve food in the form of cotyledon or endosperm, it can sustain for until the plant starts photosynthesizing. Third, inside the seed there is a young embryo. Because of the presence of seed coats, two seed coats in the seed, that offers protection to the developing embryo inside. Because seeds are in most cases, a product of sexual reproduction. During seed formation, meiosis would have taken place. So that will lead to newer genetic combinations and that will add to more variety in the population. So that is also advantageous. Now, seeds are the basis of our agriculture, right? You can't think about agriculture without seeds. Many fruit plants, fruit producing plants, vegetables, many grains and cereals, all of them are, cultivated by making use of seeds itself. So seeds are our basis of agriculture. Of course, certain types of plants you can grow vegetatively as well, but most kind of plants that we use as staple food in our daily life, most of them are, cultivated by making use of seeds. So that is the basis of our agriculture. That is also advantageous to us. And seeds, when they are released from the plant, they lose a lot of water, they become dehydrated. because they become dehydrated, because they become dormant and inactive, it's possible for us to store those seeds for a long period of time. If it were not dormant, if the seed started to germinate the moment it was released from the plant, imagine how difficult our lives would be. We would not have been able to store grains and all of these pulses in our homes. We are able to save them. Farmers are able to save them, store them until the next season before they can plant it for in their fields. They are able to store them. That is very, very advantageous to us, right? Even we can store grains like rice and wheat in our homes. We can use pulses, we can store pulses in our homes, not fearing that it will germinate the next day because of the property of dehydration and dormancy that we see in seeds. When seeds mature, they lose a lot of water because if there is excess water, it can get infested by fungi or bacteria. They lose water, they just conserve or store or preserve enough water that is just required for their survival, just required for their minimal metabolic activities. Otherwise, they will get dehydrated and lose all of that water. Because of that, pest infestation or bacterial or fungal microbial infestation can be prevented. And because they are dormant, until the conditions are not favorable for them, until there is water around them, until the temperature is suitable for them, they will not undergo germination. That is why in their dry conditions, because of their dormancy, we are able to store them for a long period of time and we can make sure that we have food throughout the year, right? So these are some advantages that seeds offer to us. Okay. Now, similar to how pollen grains have a limited viability period, I told you the moment the pollen grains are released, it can't forever retain the ability to form a pollen tube. Different species have different pollen viability during which they retain the ability to germinate and form a pollen tube. Likewise, seeds also. The time duration during which they still are capable of germinating and producing a plant, that is called seed viability. Different species have different seed viability. Some of them have been viable for thousands of years. So some examples we will learn in today's class. One is Lupinus arcticus. Okay, so this was excavated from Arctic tundra. You know, tundra region, it's super cold, it's polar almost. So over there from the snow, one seed that was almost 10,000 years old, that seed was excavated from the snow, and when suitable conditions were provided, that seed not only germinated, it also apparently produced flowers. So it had remained dormant for the last 10,000 years. It was viable even after 10,000 years. Another example is Phoenix dactylifera, which is nothing but our dates. This was excavated from an excavation site near King Herod's palace near the Dead Sea. Apparently, this also germinated after being dormant for 2000 years. So the seeds can remain viable for such long periods of time as well. Talking about the fruit, fruit is formed from the wall of the ovary. The ovary wall forms the fruit wall. Within the fruit, you will have the seed, right? So the fruit has two parts, the seed within that's present within the fruit and the wall of the fruit. The wall of the fruit forms from the ovary. Just after fertilization, as the ovule is developing into the seed, simultaneously the the ovary wall will also mature to become the fruit wall. The wall of the fruit is called the pericarp. Peri means around, carp means fruit. Okay. Depending upon the nature of pericarp, you can classify fruits into two types. Fruits that have a fleshy pericarp, fleshy juicy pericarp like an apple and oranges, mango, that is called fleshy fruits. And in fruits where the fruit wall is dry, you call them dry fruits. These dry fruits are different from the dry fruits that badam and cashew, all of that. That's a different kind of dry fruit. That is layman dry fruit. Botanically, when we say dry fruit, we essentially mean that the seeds are not covered by a fleshy fruit wall. You've seen groundnuts, you've seen peas, you've seen beans. In them, you do not find a fleshy fruit wall present, right? So such fruits are called dry fruits. Now, you can also classify fruits based on which part of the flower develops into the edible part of the fruit. In most cases, it is the ovary that will contribute to the edible part of the fruit. If it is the ovary which is contributing, then you call it a true fruit. That is the case in orange and mango, papaya, everywhere. But in some cases, like apples and strawberries and cashews, you can see that the ovary is not the part that is contributing to the edible portion of the fruit. It is the thalamus. Okay. So in such cases, you call the fruit a false fruit or a pseudocarp. Now moving on. Yeah. So this is the apple. This is the part that we consume. That the part that we consume is actually the thalamus. The part that we throw away, the hard part inside, that is the actual ovary of apple. Same for strawberry. The part that we consume, the fleshy part is the thalamus. The actual fruit are these tiny, tiny seed-like structures that you find which are called achenes. So that is the actual fruit. Okay, the fleshy part that we eat is the thalamus. Now, we have something called parthenocarpic fruits as well. You may have noticed how papayas are, some papaya varieties are seedless, some grape varieties are seedless. How is that? Fruits are forming without seeds. Fruits, if they are formed without fertilization, then seeds will not be present inside. Okay. So, parthenocarpic fruits are fruits that are formed without fertilization. They are also called seedless fruits. Many plants over the course of evolution, because of let's say not natural selection, artificial selection, have become seedless. Like bananas, the wild banana varieties used to have big seeds. Because of artificial selection by man, they have evolved into varieties which does not produce any evident seeds at all. But sometimes by spraying chemicals, you can make fruits parthenocarpic. For example, by spraying auxins, you can make tomatoes seedless. Okay. So that way, by spraying plant growth regulators, you can induce parthenocarpy in some species of plants. That's about parthenocarpic fruits. Now let's move on and talk about this mechanism called apomixis, a phenomenon called apomixis. Basically, apomixis is actually a type of asexual reproduction, but it tries to behave like sexual reproduction. You know, it is a kind of mimicry where asexual reproduction is mimicking to be sexual reproduction. You've seen mimicry artists around. There are people who try and act like Shah Rukh Khan. There are people who try and imitate Amitabh Bachchan or whoever. So, when people are trying to imitate another person, they pick up certain characteristic features from that person and try to emulate that, try to do something similar. Like, for example, if somebody tries to imitate Shah Rukh Khan, they try stammering like Kiran, right? So that is something that Shah Rukh Khan does. So if somebody else wants to imitate that person, they pick up that particular characteristic of that person and try to do the same. Over here, I'm saying that it is asexual reproduction, but it is acting, pretending like sexual reproduction. So it picks up one feature from sexual reproduction and shows it. So it appears like sexual reproduction while it is actually asexual reproduction. The character that it picks up is production of seeds. When does seed formation happen? Seed formation in plants happen only as a result of sexual reproduction in most cases. If a seed is forming, you will assume that it is sexual reproduction. But in these plants, such as Asteraceae and grass members, seed will form, but fusion of gametes only would not have happened. Even without fertilization, if seed is forming, then that is called apomixis. If without fertilization fruit forms, that is parthenocarpy. If without fertilization seed is forming, then you call that apomixis. Okay, so how exactly does the seed form here if fertilization itself does not happen? What happens here is, inside the ovule, meiosis will not take place. Egg cell is produced by mitosis itself. So the egg cell will be diploid. This diploid egg cell that is formed without meiosis or reductional division will directly develop into the embryo without undergoing fertilization. Such embryos are called apomictic embryos. Here you will not see any new variation in the next generation because it is asexual reproduction. Meiosis itself has not occurred. So this has been used by farmers. This can in fact be used by farmers. Certain farmers for certain crops rely on hybrid seeds year after year. They buy hybrid seeds from the market by paying hefty money. If at all we figure out which is the gene that is responsible for causing apomixis and try to introduce those genes into these hybrid plant varieties, you can make sure that they produce seeds even without undergoing fertilization. If you buy hybrid seeds, you sow them in your field, in your crop field, you get the yield. From your yield, whatever seeds you get naturally, normally, there the combination of characters may have changed. If sexual reproduction happens, you might not get back the same combination of characters that you desire, because newer combinations will be generated every time during sexual reproduction. If you want to make sure that the same combination of characters is retained in the progeny as well, you might introduce these apomictic genes into those hybrid plant varieties and make sure that there is no segregation of characters. Meiosis itself does not happen. So the same combination of characters will be retained in the seed and it will get passed on to the next generations as well. By doing this, farmers can save their money that they use to buy hybrid seeds year after year, and there's a lot of research that's going on in this area currently. Okay. Now, the next and almost last part of this chapter is polyembryony. It is possible that sometimes within the same seed, many embryos are present. Normally, what happens when you sow a seed in the soil, from one seed after germination, one plant will arise. There have been cases where when one seed has been sown, more than one plants germinate out of it. How is that possible? It's possible only when within the seed there was more than one embryo, right? So such cases we observe in the seeds of citrus and mango. So over here within the seed there will be many embryos. Poly means many. So the phenomenon where within a seed there is more than one embryo, it is called polyembryony. In polyembryony there will be one embryo that is formed because of fertilization from the zygote. Okay, so one zygotic embryo will be there. But along with that zygotic embryo, many embryos would have formed either from the nucellus or from the integument. So basically some cells of the nucellus or the integument will penetrate into the embryo sac and start behaving like embryos themselves. And as a result, within one seed you can have many embryos formed. That is called polyembryony. So with that, we've come to the end of this chapter. There are a lot of examples that you'll have to remember from the beginning, right? So there's examples for polyembryony, there's example for apocarpus, syncarpus condition, there's example for apomixis, there is the example for pericarp, flesh fruit, fleshy fruit, dry fruit, true fruit, false fruit, endospermic seed, non-endospermic seed. A lot of examples are there. So it's important for you to memorize all of these examples. So make separate notes for the examples alone. Keep it separately in a notebook so that you can revise them frequently and make sure that it stays intact in your memory for a long period of time. I hope this session was helpful. I will meet you yet again with another session very soon. Let me know if you understood all the concepts very well. Let me know in the comments if this helped. So I will see you very soon again. Until then, take care of yourselves, stay healthy, stay happy, keep smiling and studying hard. All the very best. Thank you.